The only thing is All right. Thank you. Okay, welcome to your first topic, physiology. I don't know how many marks this week or you can allow to on 10% of the first exam. People don't do well in it. So you should. That's the outline for today. We're already had a by today. And you can start looking for the next hour and a half. And then after that, we will have that physiology. E12 chemistry will help a lot with this, specifically understanding ions and ions and anions. It's my part of physiology is learning about story. It's a chemical story, follow along the track. So moving into it. So I've also included the yield, and you guys like that. So the top graph, I've included that. I've also decided to include a couple of ones we can learn from memorize. Just to find out. And we'll begin with the first one. Okay, neurons. What are they? So these cells are involved in communicating and gap into different parts for different reasons. Now, I wouldn't bug down too much on understanding what these are too much. What I would learn is understanding what an axon is like. So if you look at the arrow on the diagram, you can see over there, that's called axon. And the reason why that's important is that's where action potentials begin. So we'll learn what action potentials are soon. Another important thing to note are where action potentials are and their limits. Okay, so before we move into things like potentials, it's important to understand the basics of order. And that involves a lot in understanding how the concentrations of ions are physical. So if you look at intracellular, so before that, the factors that affect us are essentially your ion concentrations. But then the next question is, well, what influences those concentrations? What causes the build-up of something on the outside and the build-up of something on the inside? And that comes down to three factors. Number one, attraction of ions. Now, if you look at the diagram on the right, you'll see within the extracellular space there's a lot of sodium and there's a lot of potassium in the intracellular space. What keeps the potassium in the intracellular space is those large anions that you see. Those are proteins that are negatively charged and they will attract and all those positives in there. Likewise, they go in the extracellular space and hold sodium. Although sodium likes to move across, it can't as much because it's less permeable. However, we do see that will result in some sodium within the intracellular. You can see in the diagram over there. But the key point is that it's less compared to the extracellular. And that's something very important to understand because we'll see it's quite less in a second. So I included the summary table. That's important. I would advise you to memorize that. So sodium is low on the inside, high on the outside. Potassium is high on the inside and low on the outside. And chlorine is high on the outside and low on the inside. And we'll see the elements of those instead. Next thing we need to be aware of protein channels. Protein channels. So we have different types of channels that allow these ions to move across. The first one are leaky channels. They're selective. So each one has their own ions too. And they allow it to diffuse across the concentration gradient. So it's along the concentration gradient. Starts from very high concentrations and percent. The next one is the sodium potassium pump. That is one of the most important pumps in your body. So it will keep on coming across in semester 2, and you're going to keep on coming across that in year 2 as well. So learn that. What it does is it involves pumping out 3 sodium ions for every 2 potassium ions. And it works like that. Remember, we want more sodium on the outside, so sodium will be moving out, and potassium will be moving in. Keeping in mind that sodium is already high on the outside. So in order for it to pump out against the gradient, it's going to need to use ATP. So it's an energy active process. The next type of protein channels, voltage gated channels. these open and close based off the membrane potential, the voltage. So the important thing is potassium, it can either be open or closed. Sodium, however, can be open, closed, or inactive. And that inactive form is very important in making sure the action potentials don't go backwards. And we're going to see how that works. So the first type, gradient potentials. Something happens within the neuron, like a signal comes in or it's instructed to do something. And that will open up some sodium channels. And sodium will rush in. So the resting membrane potential of the neuron is negative 17 millivolts. And sodium will move inside. So sodium will come in, and because it's a positive ion entering the cell, that potential difference between the membrane gets less. So, another thing to know with gradient potentials is that it's very bidirectional. So it can, imagine if we go back over here, if I have a potential come down, so if I have a gradient potential starting over there, for example, this potential can go to the left and the right. And that's what we mean by bidirectional. And if enough of them accumulate, then you can get an action potential. Once you have that business. So action potentials, how do they work? Well, you can have a collection of gradient potentials, and that gradient potential, if it rises high enough, it will hit something called a threshold. So it's negative 55 millivolts in neurons. Once it hits that, it's fully activated your action potential. So what's going to happen is initially those sodium channels are going to open up. Those voltage sodium channels actually, they're going to open up. And that will result in a massive influx of sodium very quickly. Why can that occur? Because when sodium is again higher on the outside, lower on the inside. So we just then that's going to keep on continuing until it hits on positive 30 millivolts. And so once it hits that, those channels are going to close. Different sources say different things. You could see positive 30, positive 40 millivolts, or whatever it's being told by you. So once it hits those positive 40, what happens is those sodium channels close. And now the voltage gated potassium channels open. Potassium being lower on the inside, being higher on the inside, lower on the outside, resulting into rushing. And that means less positives are going to be inside the cell. And so the resting membrane potential is going to drop. So whenever we say polarization refers to negative. So depolarization, which is the first step, is making it less negative. Repolarization, which is potassium moving in, it's making the membrane potential more positive. But the potassium channels are a bit slow. That means that when something closes, it takes a bit of time for it to close. And that results in hyperpolarization. So that means too much potassium ions has actually moved out. And you'll see in that stage. But then, by the time the potassium channels are closed, remember those sodium channel and the sodium potassium channels I talked about, the sodium potassium pumps, those ones will be the things that are restoring your membrane potentials back to the resting membrane potential. So refractory periods, why are they there? So we have an absolute refractory and relative refractory. And this relies on the concept that sodium channels can actually be in an inactive state. So when inactive, they cannot be opened, they're not closed, and nothing can go through. Why is that important? Because during repolarization, sodium channels don't actually close. They go into an inactive state. But in hyperpolarization, some of them are closed, some of them are inactive. What's the relevance of that? Well, yeah, that's the refractory. So because it's inactive, it needs to be closed first, then it can be opened. And that stops the action potential from going backwards, because if you can imagine a neuron, then if I have a diagram of this. Let me see if I can. All right, let's go back to this diagram over here. So the reason why it's stopped action potential from going backwards is if we imagine that this part of the neuron, for example, is at the moment undergoing depolarization. This part. The part before it will be undergoing repolarization. So that means the sodium channels are open during this part, won't trigger an action potential into the part behind it, because that phase is in repolarization. It's an inactive state. So this makes sure that action potentials are continuously moving downwards. But in a hyperpolarized state where you have a refractory period, relative refractory, some sodium channels are inactive, some are closed. So what that means is that if you have a threshold, if you have a signal that's strong enough, you can actually cause a second action potential whilst that neuron is in that place. But it's tougher, it just needs a greater stimulus because some of the sodium channels are inactive. Okay, IPSVs and EPSVs. Well, this action potential line stuff, it happens because we want to carry a signal all the way down from the dendrite region, so the cell body regions, down the axons and through the axon terminals. But what actually triggers that response in the first place? What causes that action potential to occur in the first place? It could be an instruction from previous neuron. Now what happens is you can have excitatory, or inhibitory with synaptic potentials. So those excitatory synaptic potentials, as you can see on the diagram, what it does is it pushes the membrane threshold up. So if you have a look at here, you see how it's a -17 resting. If I have an excitatory signal come in, it's going to be bumped up. So where would that signal be coming in? The signal, for example, will be getting picked up over here on the space from a previous neuron or something. What about inhibitory? So inhibitory, what happens is it actually opens up chlorine channels, and chlorine, remember, it was higher on the outside, so it'll move in. Sodium moving in means that the membrane potential goes up. Chlorine is negatively charged. When that moves in, that will cause the membrane potential to go down. So that means it's tougher for it to be triggered. And that's useful in making sure that you don't have neurons firing you don't want them to. It inhibits them, hence the name inhibitory prosynaptic potentials. Whereas excitatory prosynaptic potentials, the objective of it is to get in a state where you can trigger action. Summation. So summation essentially refers to these EPSTs when they come, when they're triggered. So signals are being sent to this neuron, for example. Now, the way that this neuron could be pushed up to a level where it has an action potential to do two different ways. Number one neuron continuously fires off to the second neuron and tells it, get excited, get ready, keep on going until you hit that threshold. Or the second way is you have two different neurons firing together, trying to get this neuron into a position where it can have action. And that's what temporal and spatial means. Spatial, it's many. Temporal is one neuron with multiple EPSVs being fired rapidly, back to back. So what does all of this do? We'll see what all of these action potentials do downstream, but before we get to that, we need to understand the receptors. So receptors, you have two different types. You have ionotropic and ionotropic let ions in. So you can see in the left-hand side, the diagram. The sodium channels are, the sodium is moving in, potassium is moving up, it's ions moving across. Whereas metabotropic, what happens is that this, whatever that signal molecule is, it binds to the receptor and it causes a change on the other side of the membrane. And that triggers a bunch of proteins. And then that's how the signal goes down. So proteins will be triggered and then it will just carry on the signal. Metabotropics, they're slow, but the response is amplified, strong. Whereas ionotropic is quite quick because it's just ions moving across, but the response will be less. So think of where ionotropics will be more useful, and we will want rapid, short-lived movements. You know, for example, in the heart, contract, relax, contract, relax, contract, relax. You don't want to have prolonged contraction, and you don't want to have prolonged relaxation. So that's where it'd be useful. Whereas metabotropic, it's a slower but larger response. So synapses in, just electrical lines in. Now, why do we care about these action potentials? Because all of these action potentials will come down and they will get towards the end region of the neurons. And if you recall that line, it's all the axon terminals. So if you go back to this diagram, it will go all the way to the axon terminals. The potential is traveling all the way down to the axon terminals. What happens there? Well, there again you open a bunch of channels. This time it's calcium channels. Calcium comes in and it triggers a series of intracellular activities, including binding to proteins and things like that. You're not expected to know exactly what happens. Just calcium comes in, and that causes the exocytosis of neurotransmitters. So neurotransmitters are what neurons use to communicate with one another. The most common one being acetylcholine. Acetylcholine will be released, and then it will bind to its next neuron. So we call that the postsynaptic neuron. The synapse is that gap in the middle. So the neuron that's after that gap that's receiving instructions is your postsynaptic neurons. They will be the ones that will be told, okay, it's your turn now. Carrying on this message, or in this case, if you have something else being released, like an inhibitory postsynaptic neuron, it's going to tell the next neuron, be quiet, don't do anything, don't fight. And you see those within different cases. So, we go some time. I'll just explain an example of it. You're not expected to know it, but for example, in the muscle reflex, whenever we test our reflexes, what happens is imagine I'm going to put a knee reflex, and I'm going to hit a tendon over here, and my leg is going to. Now, what happens is I have my quads contract, my leg goes up. But imagine if my hamstrings were also contracted at the same time whilst my quads are contracted. Both going together, the hamstrings are weaker, so they will get pulled, and that's how hamstring injuries occur. It's when the quads and the hamstrings contract together. The quads are stronger, they'll pull on the hamstrings, and then it causes damage. So in our reflexes, what happens is the neuron that fires off the hamstring, it's inhibited. So relax. Because if you don't relax, you're going to get injured. So that's where, for example, you might have inhibitory postsynaptics. Or you may see inhibitory postsynaptics being released, being reduced in the brain, and then having some conditions where patients are shaking. Yeah, you could have Parkinson's. It's a reduction in those inhibitory chemicals. So the patient's having tremors. Or you could have Huntington's, where the patients just aren't moving around and stuff. It's because those inhibitories are not there to stop the neurons firing when you need to. But yeah, let's just add a bit of context as to why we're doing this. Not that it's useful. It's terminating neurotransmitters. So you have your neurotransmitter, it's come along, it's done its action, but we don't want it to stay there for too long. Just 1 second. Okay, I hope that's the audio is fine. Okay, I'll just say. So we have our neurotransmitter, it's come along, it's done its job, but we don't want it to stay there forever. We want to have it taken up or gotten rid of. How does that work? Well, you have 3 different mechanisms. Number 1, your presynaptic neurons, the ones that originally released it, will just take that neurotransmitter back. Just take it back. And you can see that with the diagram. You take it back up. The next thing is enzymes can break that down. And it's important that you are aware of this because that's one of the drugs that you need to know. You can target these enzymes after breaking these down. You may see the drug pyridostigmine in your lectures. That's something you're expected to know. Or the third one is that it just diffuses away. Now this diffusing away happens a bit less, but it is another way. So, there we go now. Let's see how much time. We're going pretty well, so I'll just stop here. If you have any questions so far about anything, put your hand up. We're going well on time, and I'll just come around. And if you're on Zoom, just send your questions through.
I'm just surprised.
So I learned from another senior's notes that during the relative refractory period, which is where after the action potential goes down, you can actually create another action potential. But is that correct? Is that what you guys have as well?
Yeah, so in the relative refractory period, the blue part, you can see over there. So what happens is some sodium channels are inactive and some are closed. So those closed ones, if you have a signal that's strong enough, it will open up those. But it's just, yes, you can have a second one being triggered, but it's just being the stronger signal. Okay, good. Any other questions? Yeah. Black tree. No other questions? Yes. 20 or 30 slides.
20, 30 slides. How many slides are there?
I know, she has a question.
What inactivates the sodium?
Okay, we'll continue on since no one has questions. All right. Yeah. That's lecture. Right. Okay. So spinal cord. You expected to understand some things about spinal cords. The reason why I have gone a bit more in depth for this section is because I feel like it's a bit challenging to grasp your head around these concepts without understanding a bit of theory behind it. That's at least how I felt. So if you look at the spinal cord, you know, what happens there? Well, you have a bunch of neurons coming out and they form these fibers. And these fibers travel all throughout your body. Now, one misconception that I had initially was that short neurons are these short, tiny cells that keep on synapsing with one another. And that wasn't the case. In fact, there's a neuron that starts from your brain and goes all the way down to the cell. One single long neuron. Okay? And that is a possibility. So be aware of that. So your spinal cord and your brain, they form part of your central nervous system. And that's where you have things being processed, instructions being developing from there, and you control essentially what's keeping you alive. It's really a CNS. So a bit more details on that. So some good definitions to learn is that well, these terms essentially. So what is a ganglion? A ganglion is a group of cell bodies. It's where, you know, those cell bodies, remember going back what cell bodies are? So if you look at the left-hand side, near the nucleus, the cell body is over there. So there's a lot of synapsing occurring over there. There's a lot of neurons communicating to the next neurons, and that's called a ganglion. But what's unique is that it's in the peripheral nervous system. The ganglion is something, there's a group of cell bodies in the peripheral nervous system. In the central nervous system, we call it something different called nuclei. So this is where students begin to get confused. What's the difference between nuclei and nucleus? Nucleus is that the organelle within your cell. Nuclei are those collections. Afferent, towards this, anything. So afferent nerves are towards the central nervous system. There is an acronym, SAME, sensory afferent motor efferent. You could learn that. I personally learned A comes before E, so you need to receive instructions before you send out instructions. You're not going to send out instructions blindsided. Ipsilateral just means on the same side. Contralateral means on the other side. And now that we know these terms, we're going to put them into a big picture. So, your spinal cord, biographic cross section. So, what happens is you have two regions called the gray matter and white matter. And the white matter is called white because it's a fat, and the fat is from the myelin sheath around the neurons. That is formed of fatty substance. Whereas gray matter can be unmodulated axons or cell bodies. So what happens is you can have instructions being sent up or down these areas. And then these instructions can go into different places. So if we have a look at it, we have a ventral side. So ventral is another word for anterior or another word for front. It's the frontis point. Dorsal or posterior or the back. So if you've heard of dorsal fins, that's a fin that's on the back of the fish. That's the term dorsal. You'll become more familiar with these terms next semester, but that's what dorsal root and ventral root mean. So you can have a neuron from over here in the ventral root sending out, you have a cell body over here, and it will go out. So what happens is it will go out of the dorsal root, and this region is called the horn. So the dorsal horn and the ventral horn. For the ventral horn, cell body will be there. It will travel to the ventral root. This is the ventral root over here, and that ventral root will go out. And then have a look over here. So this is what we call rami, and it splits off into two. So if we go back into this diagram, notice how there's two over here. These two join, and then what isn't shown in this diagram is that these two will split front and back. So that's the splitting front and back. If you seem a bit confused, then it's because you haven't learned this. It's probably because it wasn't a lecture, it was hidden in one of the pre-readings. So that's there. Was it hidden as a part of the pre-reading? Anyone? Yeah, it was like, all right, yeah. It was hidden as a part of the pre-reading. This will happen a lot. There was sneak things into pre-readings, and there was a sneak into these steps. So the ventral root, the neuron goes out. So remember, the ventral roots, it's signals from the central nervous system going out. So these are known as efferents. So you wouldn't have, think of it logically, you wouldn't have sensory information from your central nervous system going out. Where is it going to go to? You have another processing. So the neurons that exit there are efferents, they're motor neurons. They send out instructions, contract, relax, increase this, decrease this. They're all instructions. Whereas your afferents, which are your blue ones, they come and they bring in instructions with themselves. So they bring in, things like, hey, something's not right over here, or this temperature is rising, or I have pain over here. It goes to these afferents. So why do we care about this? Because what you need to know is, for example, so I've told you what afferents are and what efferents are. And mixed spinal nerves over here, they become mixed. So you have neurons from afferent and efferent going out and coming in, and afferent and efferent from the ventral rami. So these are called the rami, the rams. So what's the relevance? So you may receive something like, what will happen if I have a damage over here? A damage exactly over there. So if we remember that these neurons are the neurons that are bringing in instructions, the problem will be that this person will have loss of sensation in that case. Now, the next thing that you could have is what will happen if you have damage over there, at the ventral root. Think of it. If you damage the ventral root, you will still have sensation, but you can't have any actions going on. So you might have a loss of motion or loss of function. Finally, what will happen if you damage at this level, at the rami? So notice how you have both afferents and efferents. You would have a loss of both functions. Okay? So that's something you may see. But now let's talk about ascending and descending pathways. So in this, so this is not 3 yield. Let me just mention that there's a slash over there. So it's like 1 to 2. If you don't learn it, it's fine. You get through. Don't really stress. But essentially, this is how information from our body goes up into our central nervous system. So what we have is we have 2 different pathways: the dorsal column meniscus pathway and the spinal thalamic pathway. The first pathway, dorsal column meniscus, what happens is it carries information that involves fine touch, vibration, pressure, and proprioception. Proprioception, if you're not aware, it's essentially where your body is within a 3D space. You know where it is. So an example that I like to run is, for example, if you like to close your eyes, all of you, close your eyes, put your left hand out in front of you. So if you close your eyes, put your left hand out in front of you. And now with your right hand, touch the index finger of the left hand. My question is, how did you know where your left hand is, the index finger, without looking at it? And that's called proprioception. So you know where your body is in the 3D space. And it's one of the most underappreciated things ever, because what happens is whenever we're walking, we actually use proprioception unconsciously. I know where my feet are right now. I know when it hits the floor, and I know when I can pick it back up. Patients can lose this. And so what happens is they either, firstly, need to either look down when they're walking, just so they can see where their feet are. Or secondly, because they can't look down at night, they can't walk at night. Right? Because simply they don't know where their feet are at the moment in a given space of time. And that's what proprioception is. You learn in greater detail next semester, but essentially that's what it is. And it's carried through this pathway. So what happens is this first order neuron. First order neurons are the neurons that bring in the signal to the CNS. The first. That will come in through your dorsal root ganglion. So as a dorsal root ganglion, and it will go up all the way to your nucleus gracilis or medulla oblongata. You might hear it as that. A couple of different things. And it will synapse over there. Synapse meaning move on to the second order neuron. Now, this, once it synapse, it decussates. So what that means is that, remember how this was going up ipsilateral? So if the signal is coming up from the right side, initially the signal travels out the spinal cord on the right side. So that's ipsilateral, that's on the same side. But then it synapses, it goes to the other side. So it becomes contralateral now. And we know that in the sense that we know that the right hemisphere of the brain controls the left side of the body and the left controls the right. It's these neurons somewhere have to move across, and that moving across is called decussation. So that decussates on the other side, and then this second order neuron will go up all the way into your thalamus. Your thalamus, it will synapse onto the third order neuron, and it will go to wherever it needs to go. Now, the primary somatosensory cortex is this area in the brain where it receives instructions about different things: your hands, legs, face, body, trunk, anything. And wherever your body seems appropriate, it will send the signal over there and it will deal with it. So that's your first ascending pathway. Your second ascending pathway is this spinothalamic. So this checks for crude touch, pain, temperature, and pressure. So what happens here is actually that first order neuron will synapse on the level of the spinal cord. So let's say, for example, you're going to learn these things called dermatomes next semester. And that's different areas of the brain, different areas of the body that are supplied by unique nerve fiber. For example, you may have the outer parts of your arm being supplied by your C5 dermatome. So C5 or all of these, what they mean is that the nerve fiber comes off close to your C5 vertebra. You don't need to know the vertebra and fiber positionings at the moment, but just so that you're aware, if you do see it, this is what they mean. So if the instruction is coming from that side, it will synapse immediately on that level. It won't move upward at all. It will synapse and then it will decussate on that level directly. And then that will go up to the thalamus again, and then synapse onto the third order neuron. And then once again, that will go to wherever it seems appropriate in the primary somatosensory cortex. Finally, descending pathways. This is a bit more straightforward in that, now that you've received the instructions, I want to send out an action. Your action will go down. So what's unique is that 80% of these fibers are going down. They decussate at the level of medulla oblongata. Medulla oblongata is placed within the brain stem, which is positioned on the back of your head. It's quite high. So yeah, it will synapse over there, so it will decussate over there and go to the other side. Whereas the other 20% will go down, and wherever it's going to send the signal to, it will decussate at that level. So why do we learn this? the main idea here is I would advise you getting out of it is understanding what will happen if I have a damage at any position within this tract. So you may receive, so there could be a question, for example, that says, I have the damage above the decassation point on my left-hand side. What am I going to experience? And because it's above the decassation point, for example, for the descending pathway, the damage won't actually be on the left-hand side of your body, it will be on the right-hand side. And that's something you need to be aware of. Whereas if it goes below the decassation point, the damage will be on the same side. This goes likewise for your afferents. So if I have a damage, for example, let's say that's over there. So let's say that signal is coming from the right-hand side. You might be confused, why is it choosing right, not left? You will become familiar with this a lot. We talk about everything from patient's perspective. So if I imagine I have a patient in front of me, that will be the right-hand side of the patient. So signal comes in from the right-hand side, but I have a damage on the left-hand side above the decassation. So I have a damage over there. So my problem, my loss of sensation won't be on the left-hand side, it will be on the right-hand side. That's something you need to understand. Whereas if I damage it below the decassation point, I'm going to have the damage on the same side. Okay? All right. All right, reflexes. Pretty low yield, although it helps understanding it for next semester. Most of the preclinicals, like as general advice, you won't really need any clinical use. Just because you don't know it doesn't mean you're a bad doctor. But knowing it, I have found, will make your life easier in some aspects. But saying that, you can be completely fine without knowing any of this as well. But just knowing it will make your life a bit easier. Okay, reflexes. So you can have monosynaptic. What that means is that I have a signal, for example, let's say I am burning my skin somewhere. That is a traumatic signal, and that signal goes to my central nervous system. So because that's an emergency, so if I keep my hand there, I'm going to keep continuing causing damage. And that means that your body doesn't want to send it up to the brain and have a process and send it back out. You want a quick, rapid response. What that means is that at the level of the spinal cord, it will synapse onto another neuron, and that neuron will send out instructions saying, contract the arm, move it away, do something. So monosynaptic, all that means is that it only has one synapse. So what happens is your afferent will carry out that signal. At the level of the spinal cord, it will synapse onto the efferent and then go out and do the effect. So you may see a term called effector. Effector is just the thing that's defined on outcome. Most often it's the muscle. So the effector might be, your biceps contracted. That may be effector. Polysynaptic is a bit more complex. And what that means is that, yes, this afferent will go up, but then at the left spinal cord, you may see it synapse onto two different neurons, or just poly meaning multiple. So multiple synapses, you may see it synapsing onto, for example, two different neurons. And these neurons can go back to that example that I told you guys, where it sends out a signal to contract your quads, but a signal to relax your hamstrings. So that's all that means. Polysynaptic, more than one synapse. The response will still be rapid, which is a bit more complex. Whereas monosynaptic is a more straightforward response. And then, so, there's a bit of a delay, and this delay can either be explained through three different things. Number one is the time taken for the afferent signal to be sent. So, there's a bit of time until it goes through and reaches your spinal cord, and there's a bit of time until it comes back. And then there's a bit of time spent processing this information. Do I need to send it up to the brain? No. It's an emergency. Do I have to do it now? Do I have to act on it now? So these three things can explain why this small delay. So we've learned about those. Okay. Before I move on to bodily fluids, we're doing pretty well on time. Do we have any questions on spinal cord organization and things like that? No questions? All right, we'll move on. We're almost at the end, so we'll just continue. All right, body fluids. Very basics of it. So 2/3 of your body fluid is an intracellular fluid. So that is fluid that is found within your cells. That's where 2/3 of it is. 1/3 of it is extracellular fluid. So you find it outside. And a quarter of that 1/3, so 12 of your total body fluids is found in plasma. And the remaining 2/3, it's found within your interstitial fluid. So interstitial is between the cells, whereas plasma is found in the blood. So osmolarity is a number of total molecules within that solvent. So NaCl, for example, it dissociates in water and it forms sodium ion and chlorine ion. So if I have 100 moles of NaCl being mixed into this amount of water, my osmolarity will be 200 milliosmoles. Why? Because I have 100 milliosmoles of sodium and 100 milliosmoles of chlorine. Okay. Tonicity refers specifically to the things that can't cross the membrane. So osmolarity refers to the number of things in the liquid. It doesn't matter whether it's permeable, it's impermeable, it doesn't matter at all. Just the total number. Whereas tonicity refers only to the ones that cannot cross the membrane. Okay? So tonicity you may not see as your effective osmoles, because that's really what's determining where water goes. Why? Because if it diffuses across the membrane, then that solute itself will naturally establish an equilibrium balance on both sides. But if it cannot cross the membrane, then it will accumulate more either in the inside or the outside, for example, and that means water will follow those more. So remember, things diffuse from an area of high concentration to an area of low concentration. And osmosis is the movement of water specifically from an area of low concentration of solutes, or you could think of it as high concentration of water, to an area with high concentration of solutes or low concentration of water. So if you have a look at this diagram, on this right-hand side, I have more solutes, but less water in comparison to this side. So water will naturally move across the other side. Okay? Yeah. Red blood cells. They have a tonicity of around 300 milliosmoles. Now, you may see sources that say 280, 290, or 308 even. That's fine. It's around that range. Now, if I put these red blood cells in a fluid where there is low concentration of things on the outside, water will move out of the cells and into that area. So that means my cells will become shriveled. If it's isotonic, so this tonicity refers to the, remember, it refers to the things that can't diffuse across. So if it's isotonic, what that means is that water will move in and it will move out. So overall, nothing. It's net, barely any movement. Whereas hypotonic, that means on the outside, the concentration is low. On the inside, the concentration is high. I may have confused explaining hypotonic, sorry. Hypotonic, the concentration of solutes on the outside is high, and the inside of the cell is low, water moves out. Whereas in hypotonic, the concentration of solids on the outside is low, and on the inside is high. So water will move in, and it will eventually rise. So the way you can think of it is hypotonic has that O, so the cell becomes larger, O shape. Yeah, there you go. All right, body fluids, different types of water. It's hypoosmolar. It has no solutes in it, nothing. And the tonicity, it's nothing as well. So imagine distilled pure water where you have no ions. You won't have anything. So if I administer ECF, water, pure water, the water is just going to move across. Okay? There's literally nothing stopping it from not moving across. Next fluid, saline. Very important fluid, both for your preclinical studies, but also clinically. Very commonly used. Why do we use it? It's Imagine someone has been bleeding and we need to increase that volume of the blood quickly. But also we don't want to kill the cells. So how do we do that? We give 0.9% saline. Why? 0.9% saline constitutes the milliosmolar of around 280, which is very similar to your red blood cells. That means water net movement will almost be nothing. So your cells won't lyse. So if I administer that in your ECF, the sodium, if I administer in the ECF, because the osmolarity is the same as the ICF, it's fine. It will remain there. No net movement at all. All right. So it's used to increase ECF. Now, dextrose, it's a bit complex. Dextrose, like glucose, it's large, it's polar, and it can't diffuse across the membrane as easily. So initially, it will be more on the outside, the ECF will be high. But as that dextrose is being metabolized and it's being broken down, it all of a sudden becomes hypotonic. So that that dextrose will disappear, and then that fluid will move across. Okay, why? Because we broke it down as dextrose. Dextrose is no longer there. Dextrose there, dextrose not there. Yeah. And then if you look at these, the concentration on the left-hand side, remember going back to your B12 chem concentration is number of moles over volume. The moles on the left-hand side and the right-hand side are the same, but the volume is less on the left-hand side, so your concentration is higher. So water will move across. Urea, is it used clinically? No, not often, but they teach you. Osmolarity, isosmol, urea can move across the membrane, although you wouldn't really want to find it because it is a toxin. You don't naturally find as much of that. So urea, once you administer it, the urea can diffuse across, and water will just diffuse across with it, just make sure that the concentration is the same. Okay, so ECF and ICF increase. But the problem is it moves across so much, the urea, because there's no urea within the red blood cell. So if you give this to a patient, it will diffuse across so much to the point where water will move in and it will cause a lysis in the red blood cell. Okay, so it's too much. And that's me. If you have any questions, that's my Facebook, that's my email. Send me questions, but not about immunology. I'll be good. Don't send me immunology questions. But yeah, that's my part done. Now Q&A time. Yeah, let's do it.
If you got any questions, hold your hand up. Thank you. Good job. Yeah, you've got questions like on the Zoom or like you've got later?
Like here.
Okay. Then give us, see if anyone's got questions. And we'll go through those.
Okay.
Because you've literally got like 45 minutes.
If you have a question on
I
Zoom,
have
please.
questions about the no.
All right. Good morning, everyone. My name is Yang. I am also a second year, and with me is
Jaffer, good morning. Good morning, everyone. I'm Jaffer. I'll be taking you for most of the second half of anatomy.
So today we'll be going through, as Jaffer said, we will be going through anatomy of semester 1, which, as you'll come to see, compared to next term, doesn't really feel like anatomy. So in terms of the content and the structure of this next 1.5 hours, so at the start, we'll just be having a very, very short few slides on the structure of anatomy on your exams, as well as the paper 1 content and the paper 2 content. Now you'll notice on the next slide that, that there's a lot more anatomy on paper 1 than there is on paper 2. You'll also notice that the median is quite high compared to some other sections. So take from that what you will. It just means that I think overall anatomy is a pretty easy section to get high marks in to maybe make up for your other sections if you study it well. Now, unlike physiology, which is a lot of understanding, I feel like anatomy is a lot more about memorization. So also do keep that in mind. Throughout the session, we'll be going through little mnemonics and little ways for you to remember things. And if anyone has any mnemonics that they've come up with, please do come and share it with myself and Jaffa so that we can share it with everyone. So I'm sure everyone will find it very, very helpful. So on today's content list, I'll be taking you from points 1 till 3, as well as the last 2 points. And then Jaffa will be taking you through points 3 to 7. So at the start, we'll be looking at some stuff regarding cell cycle before we move into tissues, and then finally going towards skin. And I think you guys have already done some spinal cord stuff, so we'll see if we, maybe skip that bit. All right, so starting off, this is, hold on. I meant to point. Oh. Okay, so this is an animal cell. I hope by this stage you all recognize what this is. What do you need to know about this? Honestly, you just need to know the basic functions of everything. So what is the reason why they're all there? And more importantly, and this is especially for your later years. So for example, the second half of this year and in year 2, the big part is knowing what specific things, what some of the specific functions are and how that might contribute to the function of certain things. So I'll give you an example. Right, if something needs a lot of energy, it's going to have a lot of mitochondria. Now, yeah, there was another example that we literally went through last week in year 2 about how something has more endoplasmic reticulum. I can't remember why, but it shows that you do need to know that for year 2 as well. So yeah, there's that. There's a basic function for each of them. Now, I think this is also covered in biochem, so I'm not going to spend too much time on this. Okay, so on this page or on the slide, we have the 3 different types of filaments. We have our microfilament, which is also known as actin. We have our microtubule, which is known as tubulin. And we have our intermediate filament, which is, just known as intermediate filament. Now, out of these 3, your most sort of high yield ones are going to be your microfilament and your microtubule. But you do want to know the general structure of the intermediate filament as well, which is intertwined strands. Now, in regards to things like the thickness, again, I wouldn't stress too much about it. You'll see in the top right, we have the general structure of each, I'm not sure what you'd call them, but we have centrioles, flagella, and cilia. Now, a basic breakdown of what each of those are and what they do. Your centrioles are involved in the development of spindle fibers and cell division. Your flagella allows for bacteria and protozoa and sperm to swim. So that's like your little tail things. And then in regards to your cilia, hold on, give me a sec. Okay, so you also notice you'll have motile and non-motile structures there for your cilia. So if a cilia is non-motile, it's probably there for sensory purposes, whereas if your cilia is motile, that's probably referring to things like cilia within your bronchus or bronchioles or things that help move mucus. Now, the first 2, as I said, are much more high yield in regards to filaments. And yeah, that's all on this slide. Now, I think the main thing to take away from this is you do just want to memorize that little table in the top right. And once you know that, you're pretty much set for this. Also, those 2 pictures down the bottom are good to sort of know in case they were to potentially ask you what it would look like. So it helps with sort of memorizing it. Okay, so here is the cell cycle. Again, if you've done biology, you should, and also if you're in medicine, you should hopefully have a pretty good idea of what this is. So you'll notice we have 2 phases of the cell cycle. Broadly, we have mitosis and we have interphase. Now, notice majority of the cell cycle is actually interphase. And during interphase, we have the stages G0, G1, S, G2. And then in mitosis, we have the 4 stages, so PMAT as well as cytokinesis at the end. Now, the main thing to take away from this slide is that in G0, it's not really a stage. It's really just if you have any non-dividing cells, that's where it will stop. So it won't proceed any further past G0. So on this slide, we've pretty much summarized what's happening at each of the stages of interphase. So cyclins and cyclin-dependent kinases, which are your CDKs, are regulatory molecules that determine a cell's progress through the cell cycle. So think gatekeepers, which is why I've got that picture on the bottom right there. Now, the little mnemonic to remember here is DAB4221, and that pretty much links the cyclins with the CDKs. It's a really easy way to remember which is linked to what. Now notice I've put a little paragraph there on the left side that actually describes what's happening. So the part where it says transcription factors that promote the expression of S-phase cyclins, that happens at each G1, S, and G2 as well. Notice with the different cyclins and the different CDKs, it's pretty much saying, I'm not too sure, to be honest. But the main thing is you just want to know the cyclins and you want to know the CDKs. And more specifically, you'll notice there's a bit of variety. So different cyclins will bind with different CDKs. And it's the important thing to take away from this is it won't proceed to the next part of the binding unless the previous part is complete. So for example, you can't have cyclin E binding with CDK2 unless cyclin D has first binded with CDK4. So that's the main takeaway from this slide. I think this does differ slightly to the diagram on the left here. I would say the slide before is probably a bit more accurate in terms of which cyclin binds with which CDK where. Now, on the right-hand side as well is a diagram demonstrating the relationship between cyclin levels and CDK activities. And you'll notice CDK activities really peak during mitosis, whereas cyclin concentration is sort of a gradual rise up to mitosis, and then it all falls after mitosis. And so when we think about interphase and we think about, G1 and G2 and about how, the description of it oftentimes is just growth and preparation. Well, what is it actually preparing? Well, we can see that there is a big increase, or slow increase rather, in the cyclin concentration. So this is very high yield. They love to ask questions about this. And so it's good to know the differences between early prophase and late prophase. What is the main difference? Well, in early prophase, your nucleolus, which is the little thing inside your nucleus, okay, disappears, and your chromatin / your DNA will condense into visible chromosomes. Okay, so logically that has to happen first because before your nuclear envelope disappears. So just keep that in mind. Early prophase, nucleolus disappears and your DNA condenses into visible chromosomes. Everything else in prophase happens in late prophase. So that's things like your nuclear envelope disappearing, your spindles forming, your chromosomes coiling, your centrosomes, which is your two centrioles moving to the opposite ends of the cell, and then your kinetochores attaching to the spindles. During metaphase, which is the second phase of mitosis, you can see on the diagram that the spindle fibers will attach to the chromosomes. The chromosomes are positioned on the equator of the cell, which is also known as the metaphase plate, which is that line that runs vertically down the middle. Note what's happening as well with the microtubules is that the microtubules are also binding to the chromosomes at the kinetochore. And the main takeaway from metaphase, or a really high yield thing to know as well, that you are likely to get some form of a question on, is that chromosomes can be stained here. And the reason why is because metaphase is the phase where the chromosomes are the most condensed. So those bean things at the bottom of the slide are examples of chromosomal staining. All right, anaphase is the third phase of mitosis, and that is where your sister chromatids are separated and pulled to opposite poles. So the spindles are sort of contracting, and now each sister chromatid is called a chromosome of its own. It's kind of confusing I wouldn't worry too much about the semantics of it. Just know that when you separate the sister chromatids, they become their own chromosome. No, the non-kinetic core microtubules are going to be elongating the cells as they pull the poles apart. So they're not really on the diagram, but you can sort of imagine non-kinetic core microtubules at the left and right ends of the cell and sort of like pushing it away, if you will, pushing the cell apart. Telophase is the fourth step. So two identical nuclei are formed at the two poles of the cell. Your nucleoli and your nuclear membrane are going to reappear. Your chromatin is going to loosen. Your cleavage furrow, which is the sort of split down the middle, forms. And this is really where, you know, obviously the last phase of mitosis. So it's nearing the end of mitosis. Cytokinesis is the very last stage. So that's where our two cells will separate and they will form two daughter cells identical to the parent cell. And this is through the action of actin microfilaments. So you can see that what pretty much happens is at the cleavage furrow, right? The actin microfilaments are sort of like squishing it. So you see those blue things on the diagram? They're not strictly your actin filaments, but right outside the cells, you'll like sort of have actin microfilaments sort of squeezing it and creating two new daughter cells. Now, what is really important, then this is also very, very high yield, is your two, I'm not even sure what to call these things, but you have P53 and retinoblastoma. And the way that you should think about these is they're like your security guards. So they're not going to be active unless you have an issue, right? Like think about when you're lining up for the hawker, right? If you're not causing trouble, you're not going to be talking to the security guard, right? So these are only activated when there is an issue and they need to stop the cell cycle. Now, more specifically, they do have their own sort of places where they will activate, and they do have their own sort of processes. So for example, P53 will activate the transcription of something else called P21, which is a CDK cyclin kinase inhibitor. And that's going to block the transition of the cell from G2 to M. Is there any way to remember that? Not really, you just have to memorize it. The fact there is that 50% of cancers have a malfunction in P53. So yeah, fun fact. Now, RB, so retinoblastoma, is the sort of second security guard, and it acts at a different phase of mitosis or the cell cycle. More specifically, it inhibits the transition of the cell from G1 to S when active. And when it's obviously inactivated by phosphorylation, the cell is able to enter S phase, continue on with the cell cycle, and sort of persist as per usual. Now, why is this all relevant? Well, when retinoblastoma, RB, or P53 is non-functional, aka maybe you have again, going back to the example, you have a club with no security guard, you can't stop the cell cycle. So your mutations will be allowed to persist. So maybe you'll have a bunch of crazy people go inside the club, which leads to development of cancerous cells, which is maybe these crazy people inside the club. Yes. So this is really, really high yield. I would pretty much go ahead and try to know all of this stuff on this slide well, because it's very likely you're going to get a question on this. Meiosis. Now, meiosis is good to understand, but it isn't super high yield. So I wouldn't obsess with spending hours and hours trying to understand the absolute everything over it, unless you're that type of guy. More specifically, there's sort of two different things that you have to take away from it, and this also links to the next little part that I'll talk about. But it's pretty much 2 cycles of PMAT to produce 4 unique haploid daughter cells. So haploid just means half, and the more important thing is that crossing over occurs in metaphase 1. Okay, so that's illustrated in the little diagram in the top right there. Again, not super high yield, but you do want to know that it's 2 cycles of PMAT. It produces 4 unique haploid daughter cells, and crossing occurs in metaphase 1. Okay, apoptosis. So apoptosis is also known as regulated / programmed cell death. If anyone knows the guy on the top right, his name is Chubby Emu. The breakdown of apoptosis, well, apoptosis as a word is made up of apo and tosis. Apo means away, tosis means falling. So it means sort of falling away, which is kind of peaceful, which is the idea of apoptosis, is that it's normal, it's good, right? It's what helps us. Now, the opposite of that is necrosis, which, as the word necros probably represents, it's not a good thing. And we'll be comparing the differences in a second. But you'll notice with apoptosis, we have seven very, very distinct steps. And more importantly, some of these steps have labels to them. So let's go through this and break down what's going on. So if I have a normal cell and it's going to be undergoing apoptosis for any reason, and that could be stuff like viral infection, some sort of intracellular signal or whatever, right? We'll have the reasons on the next slide. What happens first is that the cell will shrink, and the cytoskeleton, which you sort of remember holds the structure of the cell, is going to be broken down by caspases. Now, the second step is that the cytoplasm is going to condense and the organelles are going to compact. The third stage is known as pykinosis. So pykinosis can be broken down into two words. So pykinose and osis. Pykinose means dense. Osis means process. So you can think of pykinosis as the process of condensing, okay, which is what the third step says. Fourth step, the nuclear envelope breaks down and the DNA starts to fragment, and that is what we call karyohexis. Again, karyon meaning kernel and hexis meaning break. So it's sort of like breaking down of the kernel, aka breaking down of the DNA envelope, which is, again, as I said, sort of fragmenting. Logically, step 3 does have to occur before step 4, okay? Because if the envelope was to break down and the chromatin wasn't to condense, you might have something go wrong there. So just the order of that is also very important. You need to know that pykinosis happens before karyohexis, which happens before the 5th step, which is known as blebbing. Blebbing is when the cell membrane buds. So what I sort of think about is, when you play Agar.io and you have a really big circle and you sort of run it into the green spiky things and they start like blebbing. Yeah, that's what's happening. Step number 6 is when you sort of get broken up into different little pieces. So more specifically, in step number 6, after you bleb, the cell breaks into contained apoptotic bodies. And then lastly, those little bits are phagocytosed or broken down by another bigger circle in the same game as you. And that is apoptosis. So again, a very good thing. Now, how is apoptosis compared to necrosis? First of all, the main definition of apoptosis, again, is regulated and programmed cell death. This is in contrast to necrosis, which is unexpected cell death. So you can see the causes there. We can very much sort of classify it. Now, notice with necrosis, causes of necrosis are things like injury, infection, cancer, poisons, inflammation, and hypoxia. And all of these causes are external to the cells and tissues. Okay, I think we can all look at that list and go, it's probably not a good thing. Whereas apoptosis, notice a lot more of it might come from inside the cell, or from things like immune cell signaling, DNA damage, nutrient deprivation, increased calcium, that sort of stuff. Do you need to remember that entire list? What I would probably just remember is that necrosis is caused by external causes. And to just remember the really common ones. So, for example, injury, infection, inflammation, whereas apoptosis can be caused by things like DNA damage, radiation, things like that. Now, again, logically, necrosis is unregulated, whereas apoptosis is regulated because it's programmed sort of being regulated. And apoptosis shrinks, the cells shrink into contained blebs, whereas necrosis, the cells just burst and they sort of release the contents of the cell, which is not good for the surrounding cells. And so that leads us to the next point in regards to damaging the surrounding cells. Last 2 points there. So apoptosis causes no inflammation, which is good. Necrosis causes inflammation, which is bad. And then with apoptosis, your cell membrane and integrity remains, whereas with necrosis, that cell membrane integrity is lost. So we have a quick question here. Which stage does chromosomal staining occur? Just take a second. This should be a relatively easy question. And once you have your answer, throw it up like a gang sign. I want to remind everyone the answers are A, B, C, and D, and E. So if you're throwing up M's, right. So yeah, I'm aware of that. Yeah, so if you're throwing up C, that's good. The answer is metaphase. So chromosomal staining occurs during metaphase because that is when the chromosomes are the most condensed. Okay, so the next one we have is gametogenesis. More specifically, we have spermatogenesis for biological males, and we have oogenesis for biological females. Now, spermatogenesis, and actually, what I would say is particularly important with this slide is the text that's on the left and the right, and also understanding what's going on in the diagram. So notice with spermatogenesis, right, we have meiotic divisions, okay? And we're forming 4 spermatids. Now, these 4 spermatids are identical. There's also a very high rate of cell death to ensure sperm quality. And this occurs in males from puberty onwards. It's uninterrupted. It does slow with age. However, it continues throughout life. Whereas with oogenesis, we have asymmetrical meiotic divisions to form 1 egg and 3 polar bodies, which is illustrated sort of somewhat in that diagram. Now, the stages in oogenesis do differ to that of spermatogenesis. More specifically, at birth, you have oocytes that are ready, and then at puberty, you have secondary oocytes. And this, for females, terminates at menopause. So main takeaways are definitely the pieces of text there on the left and the right for this slide. Now, non-disjunction events are when the sister chromosomes fail to separate, which causes an incorrect number of chromosomes. So what you have to know from this is, first of all, which one is more severe, and also how do they affect the resulting gametes. So notice with normal meiotic division, it's completely fine, right? We have our 4 haploid daughter cells, whereas with non-disjunction in meiosis 1, we'll end up with 2 daughter cells with N+1 chromosomes and 2 daughter cells with N-1 chromosomes. Non-disjunction in meiosis 2, we have 1 daughter cell with an additional number of chromosomes and 1 with 1 less chromosome, and the other 2 daughter cells are normal and fine. Now, which one is more severe? Non-disjunction in meiosis 1 is much more severe as a condition, because notice, instead of having 2 normal daughter cells, we have no normal daughter cells, which is obviously not good. Now, what happens when we have non-disjunction in sex chromosomes? These are the only 2 conditions you guys need to be aware of. So we have Turner syndrome and Klinefelter syndrome. So looking at Turner syndrome, if we have a look at the chromosomal staining there, we can notice we only have 1 X chromosome, okay? Whereas with Klinefelter, we have 2 X chromosomes and 1 Y chromosome. Now, if you struggle with remembering the order of this, what I just sort of think is Turner is a shorter word than Klinefelter, and 1 is sort of less than 3. So you can associate 1 X chromosome with Turner, and then 2 X and 1 Y with Klinefelter. Again, this is very, very high yield alongside the next slide. When we have non-disjunction in autosomes, a good thing to remember, well, maybe not a good thing to remember, but a mnemonic is pedos start young, 13, 18, and Jerry, you're a bit too excited, mate. Yeah, so pedos start young, 13, 18, and 21. This is a really good one. And since you all know it now, it's not going to be weird if someone's whispering pedos start young in your exam. So it's not in the right order either, but the P stands for Patau syndrome, and that's when we have trisomy 13. And some things that can happen there are a cleft lip, undescended testes, and malformed ears. Edwards syndrome is known as trisomy 18, and that's the most common one, although it does also have the lowest survival rate. And that leads to things like a smaller head, malformed ears, heart and kidney issues, which looking at it, you can almost sort of compared to Patel syndrome, you can see is a little more severe in terms of the necessary functions for life. So it is probably why it has a lower survival rate. And then trisomy 21 Down syndrome, I think people generally a lot more aware of, is the most survived trisomy. And generally the survival rate is a lot higher than the other two. Okay, so in terms of sources of genetic variation, we have 3 different sources of genetic variation. The first being crossing over. So that's when your alleles will shuffle and they'll create new combinations of genes that are not found in either parent. And that's what leads to genetic diversity. Now, the second source of genetic variation is known as random fertilization. So each ovum/sperm has 8 million possible chromosome combinations each. So if you do the quick maths, if you multiply them together, I think it might be 8 million squared, you get a really large amount of combinations. And so that's obviously all random. And the very last one is independent assortment. So that's related to how the homologs line up on the metaphase plate in meiosis 1. So if we have 23 different chromosomes assorting completely differently, we have up to 8 million possible assortments for every cell. So that's it for the first 3 points. If anyone has any questions, I think we'll run through it probably at the end. So I'm going to pass over to Jaffa now to talk about your tissues.
See you on the next slide. You're going to point it there as well. Okay, good morning, everyone. Like XE said, I'm Jaffa. I'll take you through the next part. So we'll just start off with epithelium. I just want to highlight that all of this like anatomy and histology stuff that you guys do in year 1, Sem 1, you guys can take 2 approaches to this. You can kind of try and just remember it solely for the sake of the exam. But I just want to highlight that this is stuff that keeps on coming over and over again. And this is the foundation that they build upon for future semesters and future years. You guys are always going to see this. So with epithelium, thinking about the functions, we've got this mnemonic for you, which is PASTS, which is protection, absorption of nutrients, ions, gases, secretion, transport, and sensory perception. Now, these functions do kind of change depending on where you are in the body, but this is a general outline. We'll go through the different types of epithelium soon. There's a bit of a preview down the bottom where we've got simple, stratified, squamous, cuboidal, columnar. We'll go through that soon. So with simple squamous, these are the types of cells that will be lining your very small blood vessels in your body. And just try to keep in mind what the function of this might be and why it's located where it is. So we've written down here that it allows for diffusion, filtration, and secretes lubricating substances. So in the context of blood vessels, you've got your ions, you've got all that good stuff trying to get from out of the bloodstream into the tissues or vice versa. Cuboidal and columnar, they generally have pretty similar function, which is secretion and absorption. They do also occasionally have other fancy good stuff on them, like your cilia, microvilli, brush borders. We'll go through that soon as well. We've got pseudostratified, usually columnar, and these secrete mucus, and it is like the primary tissue of areas such as the trachea and the upper respiratory tract. Additionally, we've got stratified squamous. Now, there's different types of this tissue type, but stratified squamous, you're thinking of your mucosal linings. So like on the inside of your cheeks or inside the vagina, stuff like that. And there's also another type of stratified squamous that's pretty important to know at this stage, which is when it's keratinized. And this is the type of things that is the outside of your body, your external surfaces like your skin. And the difference between these two is, if you think about it, like the inside of your mouth, it has a very different kind of texture to the outside, mostly to just area dependent protection. We've got stratified cuboidal, which is also protective, but this is also the major component of things like sweat glands, salivary glands, etc. We've got stratified columnar, which is present, I think, almost exclusively in the male urethra, and its function is to protect and secrete. And we've also got another special type of transitional epithelial cells, which is stretchy. So it has different shape depending on whether or not the amount of pressure being put on the outside of it. So for example, in the bladder, if your bladder is empty, doesn't need to be taking up that much space, doesn't need to be taking up that much space. But you know, if it's full of urine, you're going to need to be able to expand it and keep the urine from leaking inside your body. So there's that. So this is what I was talking about a little bit earlier with the keratinized stratified squamous. So the keratinization process basically means that the apical cells, which is the cells close to the top exposed layer, they'll lose their nuclei and they'll essentially die, and they'll just form like a protective layer all around the outside of the area that it's trying to cover. And this is important in stuff like skin. You know, you hear about your dry skin, your dead skin, all that kind of stuff. They're referring to the keratinized stratified squamous. And it kind of means that you can, like if you scratch your arm or something, you can afford to lose those first few layers without really doing any harm. And these cells are also very rapidly dividing. So you're constantly getting rid of and producing new forms of these cells. And transitional epithelium, if you take a good look at the top picture and the bottom picture, you can see that there is a bit of a difference in how the cells kind of look. And that's because they kind of change shape. So in the top one, it's probably a non-distended bladder where the bladder is empty and there's a lot of less pressure being put on the top layer. But on the bottom one, you can see that there's a lot more compressed and they look a lot more like physically pulled apart. That's because they are literally being pushed apart by the fluid or whatever it is that they're holding inside. So in the epithelium surfaces, basically, kind of, if you want to think about it, humans are kind of like donuts. You've got an outside layer and you've got an inside layer. The outside layer is covered by epithelium and the inside layer is also covered by epithelium, and you've got a bunch of organs on the inside of this donut. Donut's really like a crude way of thinking about it, but if it works for you, it works. Anyway, on the outside, you've got your protective epithelium. On the inside, a lot of the time, you do have protective, but you also have absorptional epithelia. So, you know, when you eat your food, you want to be absorbing all that nutrients before passing it out. I hope you can appreciate the donut analogy now. Anyway, throughout your digestive tract and in other parts of your body, such as your respiratory tract, you do have specialized apical surfaces, which help the cells in the area perform their function. A couple of these is the cilia and microvilli. Thinking about their functions, you know, the cilia, they are elongated motile projections that beat in synchrony via the sliding of microtubules. So yeah, guys probably haven't done this there, but like in your lungs, you know, I'm sure you've heard about the cilia. They're like constantly pushing out like pathogens, dust, all that stuff out. So it's pretty useful there. And microvilli, people do like to make the comparison between cilia and microvilli, but microvilli do have a pretty different function. Their main function is to increase the surface area of the cell, and this is to increase absorption. In a like a histological slide, you'll typically see like it's like a pink little border that you will is commonly called like a brush border or something like that. So thinking back to the cell on the outside layer, there are some specialized little funky stuff happening to help the cell perform its function. So a couple of these things that you might be looking for is a tight junction, which is at the very like top of the lateral surface, which prevents the flow of materials between the cells. So, you know, if you've got two cells right next to each other, there might be a space in between the two cells down to the basal lamina. And in some places, you really want to avoid the improper passage of ions or fluids or whatever through this surface. So you have these tight junctions to slow them down. You also have the zona adherens, which just adhere the cells together, the macular adherens or desmosomes, which is the site of attachment for the cytoskeleton via the cytokeratin filaments. And you also have the gap junctions, which is, if you have to take one of these as the most high yield, I would say it's this one. And it facilitates ion flow, otherwise known as electrical coupling between the gaps for chemical communication. Now, thinking about what you guys have learned so far throughout your first semester, can anyone tell me where like this gap junction might be especially useful? Just yell it out. Yeah, the heart. Good stuff. Anyway, on the basal surface, you've got your hemidesmosomes, which essentially just glue the epithelial cell down to the basal lamina. An important thing to note here is that the epithelial cells should never cross the basal lamina. The only time that this really happens is in cancer once it metastasizes, which is not a good thing. Okay, moving on from epithelium, we've got connective tissue. So the main functions of this is support, connection, separation of tissues, and it is found everywhere except for the CNS. It's comprised of matrix, the extracellular one, and specialized cells. So in our extracellular matrix, we've got our ground substance, which is a gel-like fluid providing nutrients and waste diffusion, and we've also got our fibers that we'll cover soon. And in addition, we've got these specialized cells, and these are kind of the cells that maintain the connective tissue. We've got our chondroblasts, which mature to chondrocytes to do cartilage, osteoblasts maturing to cytes to do bone, lipoblasts to adipocytes for like fat storage, and fibroblasts to do fibrous tissue. So just note that blast means immature, still actively secreting this matrix. And then we've got the cyte, which is a mature form of the same cell that is more involved in the maintenance of said structure. Okay, so these are the 5 types of connective tissue that you're supposed to be aware of. So you've got loose, dense, a bit controversially blood, cartilage, and bone. So starting off with loose, it consists of the lamina propria and adipose tissue. There's an abundance of ground substance and has a very flexible shape. An example of this would be breast tissue. With dense, these are the things like the tendons, ligaments, and there's 2 types of dense connective tissue. We've got parallel and irregular. So parallel fiber arrangements is so that the tensile strength is only in one direction. Now, in some parts of the body, you might not need strength in multi-directional aspects. For example, in your muscles, if you do like a bicep curl, your biceps are always going to be applying pressure in one range of motion. It's not going to like suddenly require force from another side. And your irregular dense is not as strong as the regular dents, but the advantage that they provide is that they are able to provide strength in multiple directions, like the arteries and the capsule of organs. So blood, it is connective tissue, but I kind of wanted to get you guys to think about it a little bit. Does anyone have any idea why blood is considered a connective tissue? Which one? No one? Any takers? It's okay to be wrong if you just give it a shot, you know. Which one? Yeah, point me in the right direction. Yeah, all right, whatever. Anyway, the reason it's the case is because blood is contained within an extracellular matrix. Now, it is a bit more fluid than other tissue types, but basically the plasma within your blood does act as an extracellular matrix. That's why it is classified so. With our cartilage, you've got 3 main types. This is pretty important to know, to be honest, so you do want to kind of focus in. So the hyaline, elastic, and fiber cartilage. Hyaline is the most common. It's the fetal skeleton. So, you know, when babies are born, they have a lot less bones or more bones, something like that. Anyway, they have a lot of cartilage between their bones as their bones are not fully developed. This is also present in your nose, your larynx, your ribs, trachea, and bronchi. Elastic cartilage is consisting of elastic fibers. It's very flexible, and the relevance of these is that they make up a lot of the times in your blood vessels, and this is because they are, you know, the blood vessels need that elasticity to be able to maintain their form under variable high pressures. And fibrocartilage is a combination of hyaline and dense fibrous tissues, and it's found in sites with strong compressive forces. So if you think about, you know, your spine and your knees, basically. Oh, I forgot to cover this, but anyway, in the previous slide, it says that a lot of the cartilage connective tissues are avascular in nature. So does anyone have any idea what implications this might have for healing in the area? For example, if you tear your ACL, has anyone here torn their ACL? Okay, if they haven't anyway, it takes a really long time to heal because there's a very limited blood supply to the area. So they're not getting that many new nutrients and don't have much regenerative capacity. With bone, you've got two types of bone. You've got compact and trabecular bone, which is also called spongy. Compact bone, the function of it is mainly to provide strength. Trabecular bone is containing bone marrow, and bone marrow is pretty important in stuff like blood production. The types of bone cells that you've got to worry about is the osteoprogenitor cells. So it's pretty important to actually, instead of going through all these and just reading it out to you, I think we'll just break down these words, what they mean a bit. So osteo, bone, keep that in mind. Progenitor means like generator, like kind of sounds like it. And so basically this is the one that's going to be like producing the rest of the bone cells. Blast, need to remember that that is the like an immature or like the still maturing form. Site, mature. Now this osteocyte is pretty special because it gets trapped within the bone itself and it just stays there, just maintaining the bone structure. And osteoclast, class kind of means like destroy or like cleave, if you want to think about it. And this breaks down the bone for like remodeling and like calcium stuff. So if you want to take a look at these like pictures a little bit, you can see that we're looking at some compact bone here. You can see that the osteocytes, there aren't that many of them, but they are stuck within their kind of concentric lamellae, and they just sit there maintaining their structure. In comparison to this, we've got our trabecular bone here, and you can see how it's quite different. So these like highlighted pink like little blobs, they're like the hard bits, and the white structures in between is the bone marrow. So just looking at this image, we can see that they have a very rich vascular supply in this area. So they are quite good at healing themselves. And in addition, a lot of stuff actually does go on within the bones. They're not just, something that holds you up. So they do need a relatively high blood supply. I won't go too much into the anatomy of the bone. So we'll do some questions here. So the epithelial tissue in the bladder can be classified as what? Don't make me say like throw up the gang signs for yeah. Yeah, everyone's cripping. Tehran. Okay, it is transitional epithelium. So I did say some C's. Good job on that. We're going to go into muscle now. So the functions of muscle is provide movement, maintain the body temperature, maintain the posture, support the tissues in the body. An important function is actually to act as like a sphincter to keep things in and out of. And they are also an important reserve of nutrients. So there are the 3 E's and the 1 C of muscle. So they've got the excitability, extensibility, elasticity, and contractility. Okay, this is kind of important. So you've got layers of like fibrous tissue kind of surrounding the muscle, and you've got to surrounding it at various aspects within the muscle. So you can think of it as like EPE and endo. I don't know if you guys have done too much of this, but endo, when I think of endo, I think of like blood vessels and stuff, and they're pretty small. So if I think endo, I think of like the innermost things. So anyway, epimysium, you've got the outermost layer, which surrounds the entire muscle, like everything, like that, every part of the muscle. And perimysium, which supports the, surrounds the different fascicles of the muscle. And then the endomysium, which surrounds the individual muscle fibers, so like the smallest ones. And the myocyte, myocyte just refers to a single muscle cell, basically. So myocyte, you guys might have also seen like muscle fiber, which means the same thing. There's a bunch of definitions here. So myofibrils are a bunch of parallel microfilaments of actin and myosin together. The sarcoplasm, fancy word for cytoplasm of the muscle cell, no difference there. The sarcoplasmic reticulum is basically the smooth ER in the muscle, and its major function is the calcium release and storage, which we'll get to very shortly. The sarcomere is also a very, very high yield topic, which is they constantly like to ask, what is the smallest contractile unit of the muscle? It's a sarcomere. The sarcolemma is like the plasma membrane of the muscle cell, put simply. It does have some extra functions though, such as the formation of NMJs or neuromuscular junctions. And this is due to their need to be excitability and controlled by nerves and stuff like that. The T tubules, they're basically just invaginations of the sarcolemma, and their function is to allow the spread of charge faster. So it allows more forceful contractions and more uniform contractions. You've got the termina cisternae, which is the part of the sarcoplasmic reticulum that surrounds the T tubule, and the triad, which is a T tubule and two like kind of aspects of the terminal cisternae on either side of this. So you've got some kind of general keywords down at the bottom, breaking down each of them for you. Now with the sarcomere, it is pretty important to know. I'd say it's definitely one of the more high yield topics. So just breaking it down a little bit. Oh, okay. So the sarcomere is contained within two Z lines. Okay, you've got an I band and an A band that are important. So the A band is kind of in between the two I bands, and this is kind of the center of the sarcomere. Now, the sarcomere itself, it doesn't lose or gain any like filaments, but what it does is it kind of is pulled together and apart, and it's done this through actin and myosin cross-bridging that is kind of facilitated by calcium myosin complexes. We'll go through it. There is a slide, I think. Yeah. So with the skeletal muscle contraction, you've got the action potential coming in at the NMJ, and then the depolarization traveling through the T tubule system, causing Ca2 plus release from the sarcoplasmic reticulum into the sarcoplasm. This calcium binds to the microfilaments in the cell and binds to troponin C, and this troponin C activates like some sort of cascade that forms cross-bridging between actin and myosin. And these repeated cycles of cross-bridging between actin and myosin cause contraction of the sarcomere. When the action potential has ceased, the calcium returns to the sarcoplasmic reticulum, thereby ceasing actin and myosin interactions and contraction. Okay, so looking at the histology of the skeletal muscle, it's definitely one of the more kind of accessible forms. So it is very cylindrical, it looks very nice. You can see that they're very parallel, they're very organized, and you've got multiple peripheral nuclei per cell. So just on the border of the cell, you can see like those purple stuff, which is the nuclei. And it has a very strong regenerative capacity because it's very highly vascularized. And it's got striations. I don't know if you can see them that well from where you're sitting, but there's like little light pink and dark pink perpendicular lines within the actual cell itself. And this is due to the high organization of actin and myosin. I can't remember off the top of my head, but one of them represents like the Z and the I or something like that. And something important with skeletal muscle is that most of them, or I think all of them, are under somatic voluntary control, as compared to something like smooth muscle, we'll say. You know, we might do smooth muscle first. Yep. So smooth muscle in comparison, it's elongated and it's spindle shaped. So with the cardiac muscle, with the skeletal muscle, it kind of looks like a tube. But these ones look, I don't know if you guys have ever seen like a really like an individual grain of like an orange or something, if you've ever broken it down like that, but it kind of looks like that. It's got one central nucleus, and it's got no striations or sarcomeres. It's kind of disjunct, and it's got no T tubules, and they are joining via the gap junctions. They have moderate regenerative capacity, and they are under autonomous control. So if you think about where you find smooth muscle, it's stuff like your gut, where you don't really think about it. Okay, we'll cover the cardiac muscle. Cardiac muscle, I'd say functionally, it's a lot more like smooth muscle than, it's a lot more like skeletal muscle than it is smooth muscle, but there are some key differences. So contrary to the skeletal muscle, there is branching. It's not just pure parallel lines. And does anyone know why this branching occurs in cardiac muscle? Any takers? Yep, what's that?
It's a bad explanation. When your heart beats, it gets bigger and smaller. So if it's branched, it has more space to get bigger and smaller.
I'll take that. So if you think, good job, give me a round of applause. All right. So if you think about like another muscle, like for example, your biceps, right? If you guys go like this with your biceps, you only get linear movement, right? There's no way for the biceps to like suddenly yank your arm like this, right? It's not your biceps to do that. But if you think about your heart, it doesn't really have any like super set attachment points, and it is like beating in a 3D motion. It's not the same as the linear contractions. So you do need to have those branching in order to kind of contract and relax in a 3D fashion that's needed for the blood to get pumped around the body. Going back to all of the cell attachments, we do have a lot of gap junctions and a lot of mitochondria. So the gap junctions are important in spreading the depolarization waves across the heart, and the mitochondria is basically so your heart can keep on pumping for your whole life. And they do have an abundant glycogen store to facilitate this long-term beating. So similar, has a relatively similar contraction method to the skeletal muscle, except there are a few key differences. So the first of which is that the contraction is initiated by intrinsic pacemaker cells within certain points in the heart. And within the T-tubule system, I think that there is a difference where they don't have a triad, they have a dyad. Is this true? Yeah? I think that's the case. And other than that, it is relatively similar. Yeah. Okay, so we do have a bit of a summary slide here. I think this is given to you in your lectures though. The important things to know is that the skeletal muscle is the biggest. You want to know the morphology that we just covered, so how it looks histologically. The connections. In the control, you do want to know that the heart does have this intrinsic rhythm moderated by its pacemaker cells. And you want to know that the power, the skeletal is obviously going to be the strongest. The cardiac is going to have a lifelong variable rhythm. It's not always beating at the same BPM. And the smooth is usually more sustained, slow, and like rhythmic. In terms of the blood vessels, so its functions is to facilitate oxygen exchange, temperature regulation. So, you have superficial vasodilation to get rid of body heat, coordination with the immune system via lymphatics. You have fluid regulation via filtration and absorption. And you also have pressure regulation. So, your blood pressure, all that good stuff. You've got your 3 layers from inside to out. You've got your tunica intima. Intima, like you think intima close. And this is your endothelium, single-celled squamous epithelium. that prevents sticking of red blood cells and, most stuff ideally to the surface of the blood vessel. Now, the tunica media is a muscular layer that helps alter the size of the lumen and the rate of the blood flow. And you've got the tunica adventitia / externa, so adventia, an elastic layer to help maintain the blood vessel integrity. So arteries, like if you guys haven't gotten used to identifying arteries and veins, like on a histological slide or when you look at them, a good way to think about it is that arteries are always usually going to be thicker, and they are going to have a more circular lumen, like even like outside the body. Now, the arteries carry blood away from the heart. A common misconception is that the arteries are arteries because they carry oxygenated blood, but this isn't the case, such as in like the respiratory arteries or in the umbilical artery, stuff like that. The function that they do have is that they carry blood away from the heart. And if you think about their function and their form, they are a lot thicker and they do maintain their shape. And the reason this is because they have a lot more muscle, they have a lot more elastic fibers, and that makes them a lot more able to withstand those pressures. So as you get more distal and distal from the heart, that they do get smaller due to the branching, and eventually they hit the capillaries and do their thing there. Where am I? Okay, there are two types of arteries. So there are elastic arteries where these ones are the ones that are a lot more close to the heart and they withstand more pressure. So stuff like the aorta is more of an elastic artery, as seen in the bottom left picture. And you also have muscular arteries, which have a lot more smooth muscle in their media, and their role is to increase blood distribution. And this is stuff like the radial or the brachial artery. So in terms of veins and capillaries, the veins carry blood towards the heart, and they are able to withstand a lot less pressure, because they generally operate under less pressures because they've already reached the capillaries and they've gotten through them. So the flow is a little bit weaker, and they contain these valves which ensure one-way flow. And they are thinner and more collapsible than arteries. For example, if you look at the picture on the top right hand side, you can see that the artery has maintained its circular shape, and it does have a much thicker outside border as compared to the vein, which it looks like a teardrop kind of, and it's a lot thinner. And the capillaries, they are the site of gas and nutrient exchange. The reason it's at this point is because they have a very, very thin lumen, and they allow for permeability of ions and oxygen and all that stuff. Now, the capillaries, they can be potentially even thinner than a red blood cell, and the red blood cell, due to its properties, is able to kind of fold in on itself at these points to sometimes get through. So the final parts that I'm going to touch on today is that avascular structures have little regenerative capacity due to being avascular. And the articular cartilage, such as the ACL, the capillaries are not present in these tissues that are designed for continuous compression. Like if you think about it, there'd be no point of being a blood supply there if you're constantly standing, cutting off the blood supply. And the final thing that you might need to be aware of is an anastomosis, which is a bypass which provides alternative arterial pathways. So this can be forms where an artery is linked to a vein directly without a capillary bed. And it can also be like artery to artery. For example, if you have an organ and you've got a blood supply coming in from the left and a blood supply coming in from the right, the arteries could join up together and, perforate the rest of the organ to vascularize it. And this might be an anastomosis. A good function of this, a reason behind it, is to kind of provide collateral blood flow, I guess.
All right, so these last couple of slides is all about lymphatics, the skin, and some nerve cell stuff. So this is all going to be on your paper too, and there's a total of 4 marks dedicated to this content. Now, with your lymphatics, all it is it's another system alongside your nervous system and your vessels, so your veins and your arteries. And lymph in Latin just means water. So all it is it's literally there to just drain and filter your ECF, your extracellular fluid, as well as be there for immunological reasons. So for example, recognition of foreign antigens and clonal selection and proliferation. So more specifically, what is your lymph actually made of? Well, it's a clear fluid and it's made of leukocytes, proteins, salts, glucose, fat, and water. There is no pump in the system, and there's valves throughout your lymphatic system to ensure that flow is one way. Now, your lymph nodes, which are all across your body, are your primary lymphoid organs, and pretty much that's where the lymph is emptied into the venous circulation. So the main relevance of this really moving forward is if you have any sort of edema or collection of fluid, or you have some sort of cancer, you might have enlarged lymph nodes or things like that. Now, skin is also known as your interaugmentary system. So there is 3 layers to your skin, which can be broken down into more. Again, the naming is very, very logical. So derm as a word sort of relates to skin, and the words epi and hypo I think are very self-explanatory. So epi being on the sort of outermost, hypo being below. So your epidermis is on the most outside, your dermis is in the middle, and then your hypodermis is below all that. Now, one thing that I want to clarify here is your hairy skin is not just like the top of your head. If you have really strong East Asian genes and you have no hair anywhere on your body, that doesn't mean that your hairy skin is just on your head. Your hairy skin actually covers like most of your body. So your legs, your arms, all that sort of stuff. And really your thick skin or your non-hairy skin, it tends to be the minority. So on places that are prone to abrasion, for example, the soles of your feet and maybe like your elbows and things like that. And the thing to note with your non-hairy / thick skin is that the epidermis is going to be 5 times thicker than any hairy skin, because obviously if you're going to have a lot of abrasion there, it's going to be thicker to sort of prepare for that. Now, with your epidermis, so your epidermis, looking at this diagram, it's actually a very, very thin layer relative to your dermis and your hypodermis and your fascia. Okay, but even within your epidermis, it's comprised of 5 different layers. Now, these 5 different layers, the best way to memorize this is to know that they all start with stratum, and you can use the mnemonic, come, let's get sunburnt, to remember the layers from superficial to deep. Now, this, I'm just going to preface this by saying you pretty much don't need to know this for year 2. This is something that you memorize just for your exam, and then you can forget it straight after. And the main thing is obviously you want to know the names of each of them. So corneum, lucidium, granulosum, spinosum, and basale. And also you want to know roughly like how thick they are, where some of them are present, where some of them aren't present. So for example, the second layer, stratum lucidum, is only present in non-hairy skin. And so, there's a few points there which are sort of outlined on the right-hand side. So granular cells form keratin, which is obviously important for areas where there might be a lot of abrasion. And then stratum basale is where, it's the primary site of cell division. So that's where your melanocytes are. So histologically, that's what it looks like. So you can see your first 2, so your stratum corneum, lucidum, and spinosum tend to be a lot thicker, whereas your granulosum and your basale are sort of thinner layers. And you'll notice below that you have your dermis, which contains things such as your sweat ducts and other parts that are necessary to your skin. Now, your dermis. So your dermis is the connective tissue that is directly underneath the epidermis, and it's there to support and protect the deeper layers. Now, when we look at the dermis, we have 2 different layers of it. The first one being papillary, and that is your loose connective tissue that has vascular supply. So papillae sort of means fingers, okay? And so think about papillary as in it forms fingers that poke up into the epidermis, and that's represented on the diagram on the right there, where you sort of have the papillary dermis protruding into the epidermis, if you will. Now, you also have a second layer of your dermis, and that's your reticular. And that's where you have a different type of connective tissue, more specifically your dense irregular connective tissue, and that's where your sort of thick bundles of collagen are. So your components of dermis, this is a very, very simplified diagram from Raman's notes, which are very goated, very, very helpful. And you'll notice they're labeled 1 to 4 are different components within the dermis. These are very high yield. You're most definitely going to get a question on this, if not many out of the 4. So you need to know roughly the location of each of the 4, as well as the rough function of each of the 4. Now, if you can memorize like this sort of diagram, you'll get a pretty good idea of what each of these things do. But let's run through them sort of sequentially one by one. So first of all, we have the Meissner's corpuscle. So this is found in your upper papillary dermis, and it's found inside the papillae, which, if you remember, is going to be those like finger-like projections into the epidermis. And these are known as your mechanoreceptors. Now, more specifically, these are relevant and involved in fine touch and vibration. So fine touch, the best way to think about it is your Meissner corpuscles are sort of the most superficial layer of your dermis. And so therefore, they're going to be most involved in fine touch, because you're going to have more receptors there, right? Vibration, I guess the main thing to keep in mind, that sort of reminds me of the earthquake last night, is just vibration is sort of throughout. So it's also involved with your Pacinian corpuscle as well, which is at the end, at the bottom. And so, yeah, that's it. Your Meissner corpuscle, fine touch, therefore found in fingertips. Your sebaceous glands are found next to the hair follicles, but they are found in both hairy and non-hairy skin. Pretty much they secrete sebum. And so you think of anywhere that secretes any sort of sebum, which is like a mixture of like fats and fluids and stuff, you're going to have a sebaceous gland. And obviously not on the palms and soles of your feet where you're not secreting anything. Your Pacinian corpuscles are in the deep layer. So if you have a look, it's almost at the very bottom of the dermis. And as a result, it's much more about the deep pressure and vibration. The main thing with this is you want to know what it looks like histologically, and also the general function of it. So it's more onion-shaped, and it consists of connective tissue around a nerve terminal. The last one is your sweat glands, and we can classify this into 2 different types of sweat glands. So we have eccrine, which secretes sweat directly onto the skin surface, and it's found everywhere. And obviously, like it's more common where you think it would be common. So your forehead, your armpits, and your palms. Whereas apocrine is not secreting sweat, it's secreting milky fluid directly into your hair follicles. So this is in places like your scalp, your groin, and your axilla. Okay, so we have a quick question here that we're very, very quickly going to run through. So which one of the following layers is the epidermis made up of? Sorry, that is not a question. The question is meant to be, which one of these is incorrectly named? So which one of these out of the 5 is incorrectly named? And what is the correct name of the one that's incorrectly named? And hence, what is the order from deep to superficial? So you don't need to throw up gang signs for this. Just have a think. I'm going to move on in 5, 4, 3, 2, 1. So the answer is stratum serosa does not exist. It's known as stratum spinosum. And from deep to superficial, it's the opposite of come, let's get sunburnt. So it's your basale, spinosum, et cetera, et cetera. All right, now, am I correct in saying you guys have, hold on. Yep, okay. So in regards to your nerve cells, your neural tissue develops from your ectoderm. And your neural plate, if you have a look at the diagram on the left there, as we transition from A to C, notice it sort of forms to fold that neural tube, which becomes your brain and your spinal cord, which is your CNS. Now, eventually your neural crest will migrate away from your neural tube, and that's what forms your PNS. Now, in regards to the cells involved, in your CNS and your PNS, obviously you're going to have neurons in both, but the main thing you want to differentiate with the cells of the CNS and your PNS is the glial cells. The glial cells are sort of cells there to support the functions of the neurons. And again, a lot of the names can't be very intuitive. So for example, your oligodendrocytes, the prefix oligo is referring to, sort of fat, right? So oligodendrocytes is going to be involved in insulating or myelinating your axons and allowing faster sort of impulses. Now, your astrocytes is there for general support, ion balance homeostasis. Your microglia is there for your immune response. So you can, I guess, sort of think of microbiology, maybe link that to immunology to help you remember it. And then in terms of your PNS, you have your Schwann cells and your satellite cells. So your Schwann cells are involved in myelination of the PNS, and your satellite cells are involved in protecting your neuron cell bodies. Now, I'm pretty sure you guys have run over this already, so I'm not going to run through this one again. This is just looking at your different types of neurons. And there's your spinal cord organization, which you've also run through. So, all right, so this one, which one is present in the sensory ganglia? The answer for this is the bottom right. So your pseudo unipolar neurons are involved and present in your sensory ganglia. All right, so that's all from Jaffa and I. Thank you for your time.
I can go around.
Yeah, we'll just be open to taking any questions if anyone has any.
Put your hand up if you got any. Come around with a mic. Nice. Guess they did a pretty good job. So we don't have lunch just yet. Just talk amongst yourself for like 5 minutes, then we'll be having a presentation by one of our sponsors. Who paid for your lunch? So be nice.
introduction. Okay, so we're going to be talking pharmacology today. And so in terms of what we're going to talk about, if the clicker works. Okay, so I've split it up. First of all, we'll just do a brief introduction, and then we'll talk about your exam 1 and your exam 2, and I've split up the topics into those 2 sections. So your exam 2's the bigger portion. It's got your antibiotics, which is probably the main thing you want to take away from today. And probably, yeah, antibiotics, I'd say, is the big thing you want to take away. So in terms of last year's marking, on the first exam, people got an average of 3 out of 4 marks. And then for exam 2, they got 8 out of 10 marks, which is pretty good. And so that's just showing you that even though there is a lot to get through and there's a lot to cover today, usually the questions they give you are pretty nice and they're pretty general. And so you just need to know the broad mechanisms of actions of drugs. And you also need to remember the groups to which drugs belong. And if you can do that, that's more important than knowing a name. So I've given the example up there. Rather than knowing doxycycline and all the other tetracyclines, just know the general drug class of tetracyclines, and then that will help a lot. Okay, so in terms of pharmacology, we know that it's the study of interactions of drugs with living systems. And the first 2 keywords they want you to know is selective and specific. So selective drugs act where we want the drug to act. So that's at a tissue or an organ, whereas a specific drug acts at a certain receptor. And in terms of the different types of receptors we have, we've got our agonist-gated ion channel. And so that's your nicotinic and muscarinic receptors. And so that's when a ligand is binding to that receptor and it's got an ion channel in it or a pore in it. And so the ions can directly flow through that channel. The next one is a G protein coupled receptor, and the G protein coupled receptors are for your hydrophilic ligands. And so they're going to bind to a protein on the surface of the receptor, and that's going to lead to a cascade, and that will lead to the response. Your nuclear receptors are in the nucleus, or they can be in the cytoplasm, and that's going to be for steroids and hydrophobic drugs. And then you're next to your kinase linked receptors, and I wouldn't worry too much about them, but we'll talk about them a little bit later. Okay, so in year 12 and year 12 bio and chem, you would have learned about the lock and key model and the induced fit model. The only one they want you to know about is the lock and key model, and that's specific for pharmacology. And it's just that the drug binds perfectly to the receptor. And when you increase the concentration of a drug, then there's going to be an increased likelihood of the drug receptor complex formation. And that means that you're going to have a higher response, which kind of makes sense. In terms of the 2 types of drugs we have, and the main ones you need to know are our agonists and we have our antagonists. This is just briefly an introduction. Your antagonists bind, but there's no response, and your agonists bind and they will cause the response in the end. And then a few more words, pharmacokinetics and pharmacodynamics. So pharmacokinetics is what the body does to the drug. So you can see to the left, that's the absorption, distribution, metabolism, and elimination. So that is what our body is doing to the drug. First of all, to absorb it, then to move it around the body, and then to eliminate it. Whereas pharmacodynamics is sort of your modes of action and stuff like that. Okay, and then there's different routes of drugs and sublingual, oral, and usually the sublingual one is under your tongue, and that just means it avoids stomach acid. So that's a good way to get it in if there's a poor bioavailability. Okay, so our first one of pharmacokinetics is absorption, and that's when our drugs are absorbed in the small intestine where there's a high surface area. And if we think about different properties of a drug that can affect its absorption, the first one is a pharmaceutical form, or the way that a pharmaceutical company will package a drug. So a thing like an enteric coating, entera, whenever you see something with entera, it just means gut. So if there's something has a gut coating, it means it can be resistant to stomach acids. Dispersion and dissolution, I'll get to it on the next slide, but basically that just means that if something is hydrophobic, then it's able to go through the plasma membrane and it's able to spread more easily. And molecular size, a smaller drug is more readily absorbed. Okay, so on this slide, these two images are from your lecture, and you can see that if we have a weak acid in a weak acid in an acidic environment, it's going to exist in an unionized form. And so if it's existing in an unionized form, then it's able to cross the plasma membrane, and that means it can have it can be absorbed. And then the same goes for a weak base in a basic environment. So these are about how a drug can be absorbed. And the main thing is that if it's in those environments and it has those properties, then it can be absorbed across the plasma membrane and into our body. And I've given you the pKa there. You don't really need to know much about it, but it's just the pH at which 50% of a drug is ionized. Okay, so the next one is bioavailability, and that's just the proportion of administered drug reaching the systemic circulation. And so if you do it intravenously, then that's going to have 100% bioavailability. So that is your sort of gold standard, I guess you'd say. And this is just showing ion trapping. And basically what this means is it links back to the weak acid in acidic environment stuff I was talking before. If we have a drug in its ionized form, it can't cross into another body compartment. So for example, if you think of aspirin, it's excreted in the urine. Once it's in the urine, it cannot go back into our body because it becomes hydrophilic in that environment. And if it is, then it can't be reabsorbed. Okay, so in terms of distribution of drugs, the main thing is just to keep in mind that most drugs will bind to plasma albumin, which is in our blood. And so that enables our body to transport these drugs around. And so when it is bound to albumin, then it's going to be inactive. So if you think about a patient with hypoalbuminemia, then they're going to have less albumin in their blood. They're going to have less drugs being able to bind to that albumin, and that means it can be more fastly excreted from the body. Okay, the image on the left is also taken from your lectures. And so we've got our therapeutic index, and our therapeutics index is the ratio of dose of a drug producing adverse effects. Whenever I say AAs, it's just adverse effects, to the dose producing the desired effect. And if we look at our image, we've got the therapeutic range, which is in between the minimum toxic concentration, which is the concentration that's going to produce adverse effects. And usually they're going to be serious adverse effects, because if you think about it, any drug is going to have minor side effects. For example, doxycycline is going to cause gastrointestinal irritation. And we have to have our drug above the minimum effective concentration, because if it's not, okay. And if we do have our drug above the minimum effective concentration, then it's able to work, obviously. Okay, so metabolism, when a drug is metabolized, you can see there it shows the process that first of all, it's absorbed across the gut wall. It then goes into the hepatic portal vein. And in the hepatic portal vein, it's transported to the liver. So hepatic meaning liver, and it goes to the liver, and at the liver, that is where our drugs are metabolized. And so what will happen is we have two types of phases of what's called first pass metabolism. And first pass metabolism is when we're just reducing the amount of drug that's able to go into our circulation. And we're able to reduce the amount of drug going into our circulation because our liver wants to break down it because it thinks it's toxic. So phase one makes the drug more polar, and that uses something called the CYP450 enzyme, or the cytochrome 450 enzymes, and the phase two is more hydrophilic, and so it will add a synthetic hydrophilic substance. So it will add like a glucose to a drug, and that will enable it to be excreted. Okay, so we've got our two types of kinetics you would have learnt about in terms of metabolism. We've got our zero order, and then we've got our first order. Our zero order kinetics, so the image on the left-hand side, has a constant rate of excretion. And so you can see there that you've got the artery or the vein, and it's being excreted at a rate of 2.5 units per hour. So zero order is things like alcohol and aspirin, and so it's being released at just a constant rate. Our first order kinetics, however, and they're just basically the opposite of that, is when the rate of the reaction is proportional to concentration. So when you begin to increase the concentration of a drug, then more of the drug is able to be excreted in the body. So you just need to know those two types, and I'd probably just know that alcohol and aspirin are your two examples of zero order kinetics. Okay, and again, you can see the image here. So on our left-hand side, we have our first order. You can see that as the concentration increases, and so it goes from green to red. At red, there's a higher plasma concentration, but there's also a higher rate of excretion. And on zero order kinetics, you just continually get the concentration of the drug increasing because it can only be excreted at a constant rate. Okay, so half-life's another important concept, a part of metabolism. And so a half-life is the time taken for plasma concentration of that drug to reduce by 50%, and that just makes sense. Okay, so I'm going to move on to drugs and neurotransmission now. And so that was sort of the introduction. They're just the basics, and there will be a few questions on them, but not a lot. It's more this sort of stuff, the pharmacodynamics you need to know about. So I'm sure you've memorize this, and if you haven't, you'll want to. So you definitely need to memorize that in terms of our somatic nervous system, we have our nicotinic receptors, and wherever you see a nicotinic receptor, it's either at a ganglion, and if it's not at a ganglion and it's from our presynaptic neuron, then it's acting at the neuromuscular junction. And so acetylcholine will act there. We've got acetylcholine is always going to act in our ganglia, and that's going to act on a nicotinic receptor. And in terms of our parasympathetic division, we've got muscarinic, and they're acting on muscarinic acetylcholine receptors. So muscarinic is our involuntary muscle, that's our smooth muscle. In terms of the sympathetic division, we have our norepinephrine or noradrenaline, whatever you want to call it, and that is going to act on our adrenergic receptors. And our adrenergic receptors are our alpha and our beta receptors. And so you just need to memorize that the presynaptic is our myelinated neurons. And so so the neuron before the ganglia is myelinated, and the neuron after the ganglia is unmyelinated. And the postsynaptic parasympathetic nerve is short. What that is saying is that what will happen is you will have a nerve that comes, or a nerve fiber that comes from your spinal cord that is going to go, say for example, it's going to go to the gut. What will happen for a parasympathetic nerve is it will synapse on the gut, and then it will go to a short parasympathetic postsynaptic neuron, and then that will act. Whereas in the sympathetic division, it's a short presynaptic and a long postsynaptic. Okay, this is just how we get to adrenaline. So it's coming from L-tyrosine or tyrosine, and it goes through the process to get to noradrenaline and then to adrenaline. And your rate limiting step is your L-tyrosine to L3 DOPA. Okay, so in terms of our adrenergic receptors, so this is our sympathetic response. I've given you an image up there on the left-hand side. It's from semester 2, so you don't need to know it. It's just a good way to sort of understand what's going on. So you can see that our adrenergic is always our sympathetic response. So if we have something that is, say, for example, an agonist of the sympathetic nervous system, it's going to lead to sympathetic effects. Whereas if we have an antagonist, it's going to antagonize it, so it's going to stop sympathetic effects, and that will lead to increased parasympathetic effects. So in terms of the image on the left-hand side, I know it's not this semester, but it is helpful. So you can see that in terms of adrenergic receptors, we have alpha and beta receptors. And our alpha and beta receptors act at different organs, and they do different things. But say, for example, if we look to the second row down, if we have a beta-1 agonist, then it's going to create sympathetic effects. And a beta-1 agonist is at the heart, so it's going to increase the heartbeat because our sympathetic nervous system is increasing the heartbeat. And I've just given you on the right-hand side 2 ways in which noradrenaline or norepinephrine can be removed, and that is via MAO, or COMT, COMT. And just for your interest, cocaine inhibits the COMT complex, and so that leads to excessive noradrenaline within the junction, within the synapse, and that's going to lead to excessive sympathetic effects, explaining some of the cardiovascular issues that happen there. Okay, so in terms of our parasympathetic nervous system, the way you can remember it is SLUD, so salivation, lacrimation, urination, and defecation are the ones that are our parasympathetic. And so a voluntary, as I said, our voluntary skeletal muscle is always nicotinic, but for parasympathetic, we know we don't think about our parasympathetic nervous system. So it's a muscarinic receptor, MACHR, that just means muscarinic acetylcholine receptor. And in terms of what stops it there, so I talked about COMT and the Mayo complex, which breaks down the noradrenaline. In this case, we have acetylcholinesterases, and they're going to break down acetylcholine into acetate and choline. And I've given you a drug there, which is pyridostigmine, whatever it is, and that is an antagonist of acetylcholine. So if you use that, you're going to increase acetyl in the neuromuscular junction, or within the synapse. And pralidoxamine is the other one they give you, and that reactivates cholinesterases. So this was an exam question from a few years ago, and it was, what do you treat someone who has organophosphate poisoning? And so what you would do is you'd give them pralidoxamine, because what that does is it reactivates the cholinesterases, and that's able to break down acetylcholine, and then you get a normal function. Organophosphate poisoning is just when you get that buildup through an exogenous substance of acetylcholine. Okay, so our first one of drugs and neurotransmission are our non-depolarizing blockers. And so the drug on e-pharmacology is rocuronium, and that used to be it comes from the tuberculin plant, whatever. And so our non-depolarizing blockers are competitive antagonists at the nicotinic acetylcholine receptor. If they're an antagonist, they're going to block the receptor. And so if that's the case, then you're going to have no action potentials being able to fire down that neuron. And so that's the drug there. They're the main ones that are used in terms of a blockade to cause paralysis. And the reason we don't really use our depolarizing blockers, which is on our next slide, is because when you get the drug, so in this case, suxamethonium, if you get suxamethonium, what it will do is it will bind to the nicotinic receptor, but it is an agonist. And if it is an agonist, it's going to bind to the receptor, and it's going to open, because it's a ligand-gated channel, it'll open that receptor, sodium will flow in, and the receptor will now be inactivated. But the issue with that, and the reason it's not really used, is because you are causing the channel to open, you are going to get at least one or two action potentials being fired down the neuron. And as a result, you could get some muscle twitches, a brief train of muscle twitches. And as a result, that's going to lead to pain after the surgery or after the intubation or whatever they're going to do. And so that's the reason that rocuronium is the preferred one. Myasthenia gravis is another, it's a disease you need to know about. In terms of what you need to know is the treatment of it. And just briefly, what it is antibodies binding to the nicotinic acetylcholine receptor. If you get antibodies binding to that receptor, then obviously it can't work. You can't get the neuromuscular junction firing. And as a result, you are going to get psoas, which is your eyelid drooping down, and you will get other forms of paralysis that are present. And so treatments of that is anticholinesterases. If you give them an anticholinesterase, there will be a buildup of acetylcholine within the receptor or within the synapse. And a buildup of acetylcholine will mean it's more likely to bind to a nicotinic receptor that doesn't have the antibody on it. Corticosteroids, all that does is it's reducing inflammation, and you'll learn about those next semester. And atropine, it's a bit of a weird treatment, but atropine blocks the muscarinic receptor. And so if it's going to block a muscarinic receptor, then it's more likely that the acetylcholine is going to bind to a nicotinic receptor and lead to a heightened response. But with that treatment, I think it's a bit strange. Okay, and this is just from PSP Panda a few years ago, and it's just a nice summary of what we've gone through. Envenoming and the neuromuscular junctions, it's really low yield. I didn't study for it, and I don't think we even got any questions for it. And so basically, you can see down there, you've got a presynaptic neurotoxin, a postsynaptic neurotoxin, a myotoxin. you can learn them in your own time. I don't think it's worth it. In terms of the difference between venomous and poisonous, which might come up, poisonous is when you bite something or you eat something, and that's going to hurt you or kill you. And venomous is when it bites you. And the other thing is the LD50 or the lethal dose 50. And that just means that the dose of venom that's able to kill 50% of mice over a 24, 48 hour period, which is pretty brutal, but oh well. Okay, so exam 2. Does anyone have any questions? Yes. Do you have a question?
Okay. So you were talking about like when you had that thing up and it was like parasympathetic, sympathetic, and somatic, and there's like which receptors are in the presynaptic ganglia and all that. But I don't really understand the point of all that, because I thought that like if there's a line of neurons, one of them can be like relative to another neuron, it can be presynaptic or postsynaptic. So I don't understand like how there can be such a distinction between the pre and the postsynaptic neurons.
Yeah, so in terms of do you mean in terms of like the receptor types and stuff like that? Okay, I'll go back to the slide, I'm not sure. Okay, it's just there. Okay, so in terms of, I suppose it's just something you really need to memorize, but in terms of the way you can think about it, parasympathetic is our slowing down, it's our rest and digest response. And so what that will do is you've got the longer presynaptic neuron, and you can see it's long there, and that will go to, say, for example, our small intestine, and then it will synapse there, and then it will go to the presynaptic neuron. But at all ganglia, we always have acetylcholine and we always have a nicotinic receptor. Yep. Yes. Yeah. 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 So, so there's only 2 neurons that will act. So between our CNS and then our organ will only have 2 neurons that act, and our somatic nervous system only has 1. And so that is why I'm pretty sure your longest nerve in your body goes from your spinal cord down to your big toe, and that's 1 whole cell. Yeah, yeah, yeah. It's a weird concept, I know. Yeah.
Any other questions? Who had the question?
2 questions.
Yeah.
The first one, when the drug binds to plasma albumin, you said it's inactive. Does that mean that it can't do whatever it needs to do?
That's right. So what it will do when it's bound to plasma albumin, it stays in the vasculature, so it can't leave the vasculature, and it can't bind to, say, for example, if it was an agonist of something, it can't bind. So it's just bound to the plasma albumin, and it can't do anything. Yeah.
And the second one, it says that anticholinesterase stops the breaking down of acetylcholine, but it said there that it increases it. So
I might have made an error. Anticholinesterases, what they will do, because the cholinesterase, is the enzyme. So the cholinesterase will break down your acetylcholine, and then your anticholinesterases will stop the breakdown of that. Yeah, sorry.
Thank you.
To answer your question.
Cool. Okay, I'll keep moving on. Yeah. Okay, cool. So for exam 2, you've got your more anti-cancer drugs and your antibiotics and then your antivirals. Your anti-cancer drugs, I think we probably only got 1 or 2 questions on it. It's not too high yield, but I would probably know them because there is a lot. So your classic cytotoxic drugs are the ones we've been using for ages, and they're your alkylating agents, your antimetabolites, your plant alkaloids, and your novel anti-cancer drugs are your newer ones. So in terms of our first classic, we've got a cyclophosphamide. Our cyclophosphamide, what it will do is it will create a cross-link between 2 guanine bases, and it will do that diagonally, and that basically means that the DNA will stiffen. And if the DNA stiffens, then it's more likely that it's going to break. And so in this case, all it's doing is it's going to cause the DNA to break. And as a result, it's not specific to any cycle of, it's not like specific to interphase or metaphase or any of that stuff. The next one is, okay, the next one is our hormonal antagonists. For breast cancer and prostate cancer, because it's an overgrowth of reproductive tissue, things like estrogen and testosterone are going to feed the cancer. So what our drug types are doing, or what our drugs are doing, are they're going to stop the ability of our estrogen and testosterone to be able to fuel that cancer to keep growing. So our first one for breast cancer is tamoxifen. It's a commonly used one, and it's a SERM, or a selective estrogen receptor modulator. What that means is it's just going to bind to some estrogen receptors. It's not going to bind to, for example, the cancer ones. And an aromatase inhibitor prevents the conversion of estrogen, it prevents the conversion of precursors into estrogen. So it's stopping the formation of estrogen, and the one there is anostrozole. Prostate cancer is just treating things to stop the formation of testosterone. So anti-androgens, our androgens are our testosterones, stuff like that. And our GnRH, so that hormone there, and an analog of that will be able to lower the levels of androgens produced. And you can also have glucocorticoids being used, and that's good for lymphomas. So our next one is doxorubicin, and that's an antibiotic, but it's a really toxic antibiotic. So you'd only ever be using it for cancer because it is that toxic, and it's cardiotoxic, and it can cause infertility. And it's acting to stop transcription and translation. So it's acting in the S phase. Antimetabolites, the two I've given you there are fluorouracil and methotrexate. Fluorouracil, what it will do is it will stop DNA synthesis, and just when I'm giving you the specific things, just know the overall overarching idea. So fluorouracil inhibiting DNA synthesis. A consequence of that, an adverse effect, is myelosuppression, or suppression of bone marrow synthesis. And methotrexate is another drug, and you can see the pathway there. It's inhibiting dihydrofolate reductase, and so that's going to stop the formation of DNA. And so methotrexate is probably more commonly used with things like rheumatoid arthritis rather than cancer. It can be used in cancer. And an adverse effect of it is that if you're having a kid, it can, or if someone's pregnant, it can cause issues. And they're both in the S phase. The next ones are our plant alkaloids. So our plant alkaloids can include mitotic poisons, which are our vinca alkaloids and our taxanes. So these act in the M phase, and they prevent the formation of mitotic spindle. And if you don't get that forming, it's obviously going to cause issues. And I don't know how to pronounce the second drug on screen, but what it's doing is it's inhibiting DNA gyrase, and as a result, it's interfering with the coiling and uncoiling of DNA during replication. And if you don't get DNA being coiled and uncoiled, you're obviously going to cause issues with your DNA, and so therefore you can't have that replication of DNA. Adverse effects of anti-cancer drugs, there are many, and it's really quite unfortunate. So what the anti-cancer drugs are doing are they're targeting cells of your body that can grow really quickly. And because they're targeting cells of the body that can grow quickly, they're going to affect things like hair growth, so alopecia, and they're going to target skin because that falls off and grows quickly, so skin blistering and necrosis. And there's a whole other list of them, but there's some good ones. And because of the side effects, often we use antiemetics or anti-nausea tablets, and we can also use erythropoietin, and that will form red blood cells, because if we get bone marrow suppression, then we get an inhibition of red blood cell synthesis. And so as a result, EPO is a good way to combat that. Okay, so for our new drugs, there's your monoclonal antibodies, and a lot of the monoclonal antibodies, or all of them, will end in MAB, so monoclonal antibody. And so I've given you three there, which you need to know: rituximab, trastuzumab, and bevacizumab. So your rituximab triggers B-cell lysis. Your trastuzumab, which is Herceptin, and that binds to HER2, which is overexpressed in breast cancer. And then your bevacizumab binds to vascular endogrowth cell, and as a result, it's going to stop the formation of angiogenesis or new formation of blood cells in a new formation of vasculature within a tumor. And here's some more ones for you to learn. These ones aren't too high yield though, so I'll put them on a separate screen. But you've got interferons, IL-2 checkpoint inhibitors, protein kinase inhibitors. They all kind of make sense. They're all a part of the immune system. If you stimulate the immune system, you're going to have higher chance of inhibiting the cancer. The protein kinase inhibitors will work in the cell and they will stop ATP synthesis. Okay, and drug holidays are an important part of cancer treatment. So drug holidays, if you give the treatment, what you need to do is then have a break. And the reason for this is because, as I showed you before, there are so many side effects and adverse effects of anti-cancer drugs that it can cause quite a physical, but also a psychological harm on the patient. So you want to give the patient a break from adverse effects. And it also allows for cells to go into new phases of the cell cycle. And we saw there that drugs can act in different phases of the cell cycle. So your mitotic alkaloids only act in the M phase of mitosis. So therefore, if you give them the drug and it doesn't work, you take them off it, you go on a drug holiday and then you give it again. Well, that cell's probably going to be in the M phase and it's probably ready to be targeted by that drug. The other two words you need to know about are the tumor cell sanctuaries and dose exhaustion. Tumor cell sanctuaries is exactly what it says. It's a sanctuary for tumor cells to grow. So a good place for that is in the brain where drugs can't pass over the blood-brain barrier. And so in that case, the management, MX, the management is radiotherapy or surgery. And that's also why we struggle in finding treatments for and curing things like brain cancer, because they're in these tumor cell sanctuaries. And dose exhaustion, the max therapeutic dose, it's when it's reached. And so therefore the patient's response is not, or so you give them the drug, the max therapeutic dose is reached and you haven't had any sort of advantage of using that drug. So you might need to look to go into other drugs or doing a radiotherapy. Okay, antibiotics is the big one that you need to know about, and it's the big one that you will need to know going forward, and it comes up pretty much in every semester from now on, so it's good to know. So in terms of antibiotics, we can have two types of treatment. We can have directed and then we can have empirical. Directed is when we know the bacteria, so they've cultured it, they've done a microscopy, culture and sensitivity. And empirical is unknown bacteria. So that's sort of when you go to your doctors, you've got sinusitis and they just give you doxycycline, for example. Prophylactic is to prevent infection. So things like doxycycline when you go overseas to a malaria country. And bactericidal, bacteriostatic basically says it in the name, cidal to kill, static to stop. So yeah. In terms of the different ones we have here, we've got our time-dependent antibacterials, and they're more effective when the drug concentration is kept above the minimum effective concentration for longer. And that's something like penicillin, and I would probably remember that penicillin is a time-dependent antibacterial. Our concentration-dependent antibacterials, we have to maximize the concentration. So it's not about time, it's about maximizing concentration. And this achieves more bacteria deaths, which is ideal. And the post-antibiotic effect is continued suppression of bacteria after antibiotics have dropped below the minimum effective concentration. Okay, so this one's from your lecture as well. It's just showing you the minimum inhibitory concentration, which is effectively our MEC, our minimum effective concentration. And then our MBC is the minimum bactericidal concentration. So once we've reached the MIC, we've got our bacteriostatic, we're stopping bacterial growth. And then once we've reached our MBC, we're now killing the bacteria. Okay, so antibiotics, we've got a few to get through. So first of all, I'll start on the right-hand side of the screen with our cell wall layers. So we've got our beta-lactams or beta-lactams, and they're inhibiting transpeptidases. So they cross-link linear peptidoglycan polymer chains within the cell wall, and we're inhibiting that. So things that will do this are our penicillins, so our amoxicillin, and the major side effect, or not an adverse effect, but the major issue with the use of these is a lot of people are allergic to them. So that's probably why, the only reason why you wouldn't use it as a first-line treatment. Our next one are our cephalosporins, and these are cephalexin, cefuroximine, stuff like that. And the adverse effect is that they can be neurotoxic, and things say different things about whether you should use this with a penicillin or allergy or not. But generally speaking, because they've got the same mode of action, you wouldn't use it with a penicillin allergy, but probably just know the adverse effect is neurotoxicity for that one. Our glycopeptides are our mycins, not our rhomycins, but just our mycins. So vancomycin is the one you need to know. And they inhibit transglycosidases, and that is the synthesis of polymer of glycopeptides. So we're stopping that. And they're effective against gram-positive bacteria. And so the way I remember is I think vancomycin and ambulance van, it's got a red cross. So it's positive, like a plus, and red. So red man syndrome is the major side effect from that because it's a vasodilator. It can also be ototoxic and nephrotoxic, and it's good for methicillin-resistant Staph aureus and Clostridium difficile. In terms of anti-metabolites, these are bacteriostatic. So these are our sulfonamides. And what they will do is, in the formation of DNA, we've got a few steps. We go PABA, and then we go to folate, and then we go to DNA. And our sulfonamides, which is the drug sulfadiazine, inhibits PABA or PABA to folate. And as a result, it's a good drug to use. But as a result, because folate is so crucial for a baby in utero, it can be contraindicated in pregnancy. And the other drug there is trimethoprim, and it inhibits dihydrofolate reductase. So it's the same mode of action as our methotrexate, which is used in rheumatoid arthritis, and it's used in cancer. Methotrexate you wouldn't use for someone who has a bacterial infection. Trimethoprim you would, but I've given it to you there just so you know it's the same mode of action. And trimethoprim's the one you'd use for UTIs and stuff like that. Okay, so protein synthesis moving forward, we've got now it's more specific to subunits of rRNA and stuff like that. So I'll start with our aminoglycosides. What they will do is they're causing a misreading of mRNA. And if they're causing a misreading of mRNA, they're going to stop, or they're going to stop that bacteria able to reproduce. But what it will do is it's an irreversible, what it does is it irreversibly binds to the 30S subunit. And a good example of this is gentamicin, but it can be nephrotoxic and ototoxic. Oto just meaning ears and nephro meaning kidneys. Tetracycline binds to the 30S subunit, and it will inhibit the binding of tRNA to the mRNA ribosome complex. The difference between aminoglycosides and tetracyclines is that tetracyclines binds reversibly. And the good example of that, and the common one that's commonly given, is doxycycline. The issue with doxycycline is there are heaps of different adverse effects. So you can see there with that person's teeth, they've had their calcium's been chelated. What that means is their teeth become yellow, permanently stained. There's also gastrointestinal tract disturbances. You've got a sensitivity to the sun. Your kidneys and your liver can be toxified. They can have issues. And you shouldn't give it to someone under the years of 8 years old or pregnancies after 16 weeks. The reason for that is because what these drugs do is that in chelating calcium, they're going to stop and inhibit bone growth. And as a result, it's going to be bad for a kid trying to grow up or bad for a baby. Our macrolides on the right-hand side will bind to the 50S subunit, and they inhibit translocation, and they're reversible. And these are our rhomycins. So our clarithromycin, our azithromycin. The adverse effect of that is gastrointestinal tract disturbances. And you're probably getting the idea now that all these antibiotics are going to cause gastrointestinal disturbances. The other things they do is ototoxic, and they can cause funny things to happen on an ECG, so they're cardiotoxic. And chloramphenicol is the other one, and that inhibits peptidyl transferases of the 50S subunit. So they're inhibiting the ability of the amino acids to translocate between tRNA, etc. Just know that what it's doing is it's stopping protein synthesis, and it can cause something called Gray baby syndrome. I know there's a lot of modes of action to memorize here, but what I would do is I would just go, if I go back to this screen, the ones on the right-hand side, really just know that they're stopping cell wall synthesis. The ones on the left are interfering with folate production. This whole next slide is simply just all the cell wall synthesis stuff. So, protein synthesis. So just know that these drugs are affecting protein synthesis. And then the next slides I'll go through now. So we've got on our right some more drugs. We've got fluoroquinolones, which is ciprofloxacin, and that's inhibiting DNA gyrase. And so these affect mainly gram-negative bacteria. Our nitroimidazoles, so metronidazole is a good example of that drug. It's toxic to DNA production. And so the way I remember this drug is metronidazole, like metro trains. Metro trains are metallic, so it's got a metallic taste in the mouth. And metro trains obviously don't breathe, so it can only be anaerobic bacteria. And gastrointestinal disturbances again. Our last one is rifampicin, and rifampicin inhibits RNA polymerase, but it can cause red urine, as is shown in the sample, which is lovely. And so for the antimicrobial creed, you need to know it, but because it's multi-choice, you can pretty much just look at it and know. But I've given it to you there in case you're interested. So I'm not sure if you need to know about tuberculosis treatment or not, but it's here for you. So the way you can remember it is the acronym RIPE, rifampicin, isoniazid, pyrazinamide, and ethambutol. And I've given you their side effects, their mechanism of action. But the drugs that deal with tuberculosis, as you know from your microbiology, tuberculosis is a mycobacterium. It's got mycolic acid in its cell wall. So for isoniazid and for ethambutol, they're going to stop mycolic acid being incorporated into the cell wall. And as a result, you can't, or the cell will die. But I'm not sure how much you need to know about them. So I've just given you them there if you're interested or if you need to know them. Okay, so moving on to antivirals. In terms of our antiviral drugs, I would say that your antibacterials are much more high yield, and I would be focusing much more on your antibiotic drugs, because your antibiotic drugs are given far more often than your antiviral drugs, which are specific for immunocompromised or elderly patients. And it can also be the extremely young. But you would only be given these drugs if someone is immunocompromised and needs the extra help from an antiviral drug. And all our current antivirals are virostatic, so they're stopping the virus being able to replicate. They're not actually stopping or killing the virus itself, because we know we can't kill something that doesn't live. So for the flu, the ones you definitely need to know are our neuraminidase inhibitors. Neuraminidase, you can see it down in the bottom left-hand corner of that image. A neuraminidase is an enzyme that's able to cleave off a flu virus from the rest of our cell. If we inhibit that, the virus will just remain stuck to our cell, and then as a result, it can't be cleaved off. And so that's probably the best way we have at the moment of treating the flu with antiviral drugs. And these, or the main ones you need to know, are oseltamivir and zanamivir. They're the two main ones. And the adverse effect, again, is gastrointestinal disturbances, but usually it's pretty well tolerated. The next one is for the herpes simplex virus or varicella zoster, so chickenpox and HSV. And this is our acyclovir, and it's inhibiting viral DNA polymerase. Okay, so for our retroviral therapies, when we come in with an RNA and we put DNA into a cell, so a good example of this is our HIV. We've got a few different therapies we can use, and so I've listed them there for you. I mean, you should know them, but if there's a lot. And so they've got the nucleoside reverse transcriptase inhibitors. They're inhibiting the enzyme transcriptase. You've got a non-nucleotide reverse transcriptase inhibitor, so one that's not a nucleotide. You've got protease inhibitors, so stopping stopping an enzyme which break down proteins. And then you've got integrase inhibitors, so you're able to stop genetic material being transferred into the DNA. Okay, so for HIV, the main things you need to take away, and this was in an ICL if I recall correctly, you've got your pre-exposure prophylaxis, which is taken in order to prevent a HIV infection. They're your pre-exposure, and so examples are tenofovir and that one, and it's an NRTI and it inhibits viral transcriptase. Our PEP or our post-exposure prophylaxis, when you take HIV medicines, and it's usually within 72 hours after an exposure or possible exposure to HIV. And the last one is, how do you prevent it going from mother to child? Use zidofudine, which stops the vertical transmission. Okay, for protease inhibitors, there's ritonavir, and that prevents viral replication, and it slows the metabolism of other HIV drugs, and it can slow the metabolism. And the adverse effects of that are cardiac abnormalities, hepatotoxicity, and gastrointestinal effects. And then the last one, before I think I'm done, is our antifungals. And so these are fluconazole, and this is the main one you need to know. And what it's doing is it's inhibiting fungus-specific cytochrome P450, which is the CYP450 that I showed you before. CYP450, as we know, is important in the metabolism of a drug, in first-pass metabolism in phase 1. If it inhibits that, then it's going to mess, first of all, with our metabolism, so that's a big issue. But it's also going to inhibit the fungus-specific cytochrome P450, and so that's going to stop their cell membrane synthesis, and it will lead to their death. And it can be topic or oral, and it's good for, or it's broad spectrum, and it's good for candida, so candida infection of the skin, or dermatophytes, which are a fungal infection of the skin. And these can be topical or orally taken. Cool. I'm going to do questions from past exams, and then if anyone has any questions, I can take them, or you can come up at the end. It's whatever you want to do. Okay, so here's our first question. This is from a few years ago, and this is our venoming stuff, and I'll give you 10 seconds for a question. Okay. Sorry?
No, 10 seconds, just get through it, yeah.
Just get through them?
Yeah.
Okay. Okay, the answer was C. You want to apply a pressure or a mobilization bandage. The reason it's not tourniquet is because you can stop blood flow, so it's a mobilization bandage. The next question, which of the following does not have a role in the treatment of snake envenoming? Again, it's the same thing. So tourniquet is not what you want to use. The next one, an anti-cancer drug group that acts by interacting with an enzyme's ATP binding site to disrupt cancer cell growth is A. We know it's not A, we know it's not B, it's not C because that's dealing with our immune system. A monoclonal antibody is the rituximab and stuff like that, so it's not that, so therefore it must be E. I'll give you 5 seconds for this one. Okay, for that one, as I said, MOA and COMT, they're involved in the degradation of noradrenaline. And cholinesterases for acetylcholine. Which of the following is not a reason to have a drug holiday? It's C, because we know we can't access tumours in a tumour sanctuary. They're across the blood-brain barrier, for example. Which of the following characteristics would most likely result in an absorbed drug remaining in the plasma? C and D. High plasma protein binding, it binds to the albumin, and there's a slow renal excretion. That means it can't be excreted. The next one, which of the following drugs is bacteriostatic? It is our erythromycin. It's stopping protein synthesis. Yep, so acyclovir inhibits DNA polymerase, and the others I've given to you there, what their mechanisms of action are or what they treat. Which of the following, this is kind of an easy one, the 4 phases of pharmacokinetics in order, we know it's going to be A. Okay, this one is a common question and we got asked it, so you might be asked it as well. Okay, for this one, the flu has 8 ssRNA, single-stranded RNA segments. And yeah, you can go through the others, but they're wrong. The reactivation of acetylcholinesterase following inhibition by organophosphates is best achieved by the administration of, and we went through this one before. Cool, so pralidoxamine will reactivate acetylcholinesterase. This one's about trastuzumab used in cancer therapy. Okay, so trastuzumab is Herceptin. It's common chemotherapy for breast cancer patients. This one is, Yep, so B for this one. Obviously, it's going to kill bacteria in the gut, and that's going to lead to infections. And one of the big infections it can cause is Clostridium difficile, but you don't answer that. This one's about folate synthesis. Okay, so this is C. Our trimethoprim stops dihydrofolate reductase. And our last question, I believe, adverse effects of antifungal flucanazoles include. Okay, so for this one, flucanazoles we know is specific to CYP450, and so it can inhibit our own CYP450, so that can lead to effects on drug metabolism. Okay, does anyone have any questions?
Hi, sorry, can I just confirm that chloramphenicol is reversible? Because I remember learning that it's reversible, not irreversible, and Google says that it's reversible.
Oh, thanks. I can check that one. Yeah, so the question was for chloramphenicol, whether it's reversible or irreversible. I've put in the slides it's irreversible, but I'll check it and I'll update it if it is reversible. I'll check in a textbook. Yeah, thanks for that.
More questions?
All right, if there's no more questions about this topic, is there any questions about any of the other topics we had today? Med school in general, any questions?
Medea is here, so
Yeah,
he'll be keen to answer any questions you have.
he's very down. No? Oh, yep. Go for it.
So in one of the slides, it said that P53 inhibits G2 to mitosis. Is that right, or is it G1 to S phase?
G1 to S.
G1 to
Yeah,
S
yeah.
phase? Okay.
Medea?
Yeah, sorry about that. That is an incorrect slide from our previous presenters. Don't blame me, please. Anything else? Alrighty. Well, that pretty much finishes it up for our sessions today. Thank you for coming. Tomorrow we've got a couple more presentations, so make sure to be there. There won't be any pizza, but there'll still be learning, which is arguably better than pizza. So thank you for coming, and we'll see you all tomorrow. If you got any more questions, feel free to come up and ask us.